Okay, hello everybody. I'm Marcela Cornejo. I am from Pontificia Universidad Católica del Paraíso from Chile. And we are in this cruise since October 11, I think. And this is the third or fourth dive. And we are in front of El Quisco in central Chile. And we are at 393 meters depth. And we are looking for the bottom organisms and environment. And it's amazing that the oxygen here is a little bit more than 1 ml per liter, 67 micromolar. It's quite low, but not so low than we observe in the water column. This region has an oxygen minimum zone, very strong. Today we did a CTD and we record the oxygen from the surface to the bottom, and we measure 0.2 ml per liter, so it's quite low than now. And I want to tell you that we have many people here. In the controls are Tyler, Mike Leaf, and Doug, and Juliana, and also is Scott, Adam, Jeff, and Maritza. And maybe Scott, do you want to introduce yourself?
Sure. Hi guys, I'm Scott Wyman from Woods Hole. I'm on this ship running a methane sensor as we explore these cold seep areas. So the SAGE methane sensor is my kind of ROV instrument on this cruise, and I'll pass it over to Adam.
Good morning, this is Adam Skarke. I'm a researcher participating in this cruise in the Falkor 2. Today I'll be assisting my colleague Jeff Beeson here as we conduct a multi-beam mapping survey of the seafloor. This is a sonar system that allows us to create a very high resolution, a very detailed map of the seafloor around these seep sites.
Thank you. Jeff, do you want?
Yeah, hi. Jeff Beeson from Oregon State University and affiliated with NOAA's Pacific Marine Environmental Lab. And as Adam just said, we'll be tag teaming on operating the M3 multi-beam sonar mounted to ROV Sebastian to get high resolution maps of the seafloor and image any bubble emissions that we may have missed yesterday. We're getting the ROV to a good altitude before we start running some survey lines. And then a little later in our shift, or near the end of our shift, we'll be handing it over for more on-bottom sampling and exploration video transects of the seep sites.
Hello everyone, again, Maritza here. I'm marine biologist in this cruise, finally. I work with Seep, and in this cruise I work with sediment with Praxedes Muñoz from Universidad Católica del Norte.
Super. So I will talk in Spanish sometimes, maybe most of the time. Bueno, estamos a casi 400 metros de profundidad y es impresionante la cantidad de partículas y organismos pequeños que hay en la columna de agua. Es totalmente diferente a otros lugares que son más oligotróficos y las aguas son más claras, sin tantas partículas. Y bueno, esta es una de las regiones más productivas del mundo, y aquí a 400 metros de profundidad vemos cómo esa productividad se expresa. Y el bombeo de carbono que secuestra carbono desde la superficie y lo lleva al fondo, aquí se refleja claramente. Bueno, otra cosa súper impresionante es este equipo, el Sebastian, que es capaz de, bueno, con los operadores, por supuesto, es capaz de obtener muestras, seleccionar lo que nos interesa y poder llevarlo a superficie y donde nosotros podemos estudiarlo. Bueno, como les mencionaba antes, ya este es el tercera o cuarta sumersión de este robot submarino, y hemos observado muchos organismos, bueno, algunos que conocíamos. Estamos sobre un Seep, que es un lugar en donde se produce metano por la descomposición de la materia orgánica. Todas estas partículas u organismos, cuando mueren, se van descomponiendo en el fondo, y como es tan alta la productividad, se van descomponiendo, produciendo metano cuando están más enterrados. Cuando ya no hay oxígeno y ya otros procesos ya no ocurren para la descomposición de la materia orgánica, entonces se genera metano por la descomposición de esta materia orgánica, y eso genera burbujas que salen a la superficie, y hay muchos organismos que viven de esa producción de metano y también de azufre, y se genera toda una comunidad quimiosintética que abastece a otros niveles tróficos. ¿Sí? Bueno, a medida que vamos viendo los
Camarones también hemos visto, y unos pequeños peces en la columna de esta.
Sí, hay mucho ictioplancton, ¿no? Ictioplancton, que son los primeros estadios de los peces. Y ahí podemos observar los pequeños que tienen movilidad y se van moviendo ahí frente a la pantalla. Bueno, otra cosa interesante de aquí es la temperatura. Tenemos 7,96 grados Celsius. En superficie, cuando hicimos el perfil con CTD, el CTD nos permite medir la temperatura, la salinidad, bueno, oxígeno, también la clorofila a través de la fluorescencia, y en superficie teníamos esta noche casi 14 grados Celsius. Y en un CTD anterior, el día anterior, teníamos en superficie 11 grados Celsius. Mira, ahí vemos un tenóforo. Un tenóforo, y vemos ahí sus pequeños tentáculos, no sé cómo se llaman, Maritza. Bueno, uno de los procesos que mantiene esta alta productividad es la surgencia, que es la que nos lleva a muy bajas temperaturas en superficie, a los 11 grados Celsius que les mencioné, que es el transporte de aguas desde las capas subsuperficiales hacia la superficie, y cuando lo hace llega a la superficie cargada de nutrientes, y estos nutrientes entonces son utilizados por las microalgas y se generan los blooms o proliferaciones de microalgas, que son la base de la trama trófica y que sustentan toda esta comunidad que vemos aquí en la columna de agua.
us started?
We're ready.
We're ready.
Okay, well, we'll start logging.
Just tell us
with
which way,
the
how you want us to go.
Maybe Adam, just when we start logging, get it on the C log.
Yeah.
Near the M3 button. We can, there's the record.
So, yeah, we'll record, that's the raw, and then the export is the all, so you want to trigger them together.
Alright. Click there.
Yep, exactly.
Alright, I am starting to log.
How fast do you want to go?
Did you have a preferred speed or a max speed you don't want to go?
I think faster is the better, as long as you can stay online or pinging so quickly that there's not really any concerns about not getting enough coverage or enough sound things.
Alright, we're moving. At point 3.
Bridge ROV.
Alright,
Morning Al. You got an updated waypoint.
right up, right north.
South.
Okay, it's still to the north from me.
Let me push it back up there. It should be south to FKT.
Okay,
I see it on his screen. How it didn't update, I'm not sure.
Yeah, hay una medusa, ¿no?
That's
and
where.
it's moved now. Head in a sec.
Cheers. And just to give you a heads up, we're just going to be mapping like north, south, south, north.
Yeah. What sort of speed you'd have heard of?
We're trying to make 0.3 meters per second with the ROV, so I think that's what, about 0.6 or 0.5.
Alright, so for some reason we returned back to the starting point, so I'm going to send it again. Okay. I don't think you can start a new line.
Okay. We can start doing it.
Cool. Alright.
Sorry about that. Might have been when I pushed that waypoint.
So you talk about.
There we go, on the move. Yeah.
That's probably exactly
Okay.
right. Sorry.
But when you do a new line,
We need to talk about.
Yeah.
Should I turn up to the,
I'll
just turn,
Oh.
can it turn back on the line?
yeah, they just turn back on the line.
Should probably talk about start.
Yeah.
Start line, or inline start line.
And then I find this one's it's top down.
Yeah, purpose.
It's whatever you want. I just get confused as well.
Sí, seguimos avanzando. ¿Y no vemos el fondo? Will you go to the bottom? Will
No, we're going to stay at this depth, and we're just going to go in a straight line.
Ah, okay.
So you have a very fun narration as
Do you know about jellyfish?
you can talk about rain snow. For the next time.
Yeah. Marine snow. Pero ya hablé de la marine snow.
Yeah.
Sí, parece lluvia. Lluvia de partículas y organismos. ¿Y qué, Maritza, hay bioluminiscencia?
No, o sea, yo no he visto, pero los de afuera igual.
Ah, pero eso que se ve en brillante, ¿no?
Son peces que como hacen este contraste de
Con las
la
camas.
moviéndose, se ve como
Muchos pececitos.
What's the CPM? It looks like C4.
Bueno, el rover está iluminando todo porque acá a 400 metros no tenemos luz. Seguimos navegando. Y a veces aparecen unas medusas. Como viaje por las estrellas, ¿no? Sí. Where is Chewbacca? No puedo. Pasó ayer es que sacamos con el Sebastián varios testigos de sedimento que se sacan con los push core y estos sistemas están cargados de azufre y llegaron a superficie con bastante olor a azufre. Sí. Lo que quiere decir que están muy reducidos. Algunos parecen súper rápidos. Maybe. So yesterday we saw bubbles. No? We saw bubbles yesterday, no? From the sea.
Yes, so yesterday we didn't see a continuous bubble stream from the seafloor, but we saw a couple points where, as the ROV was going over, we saw a little lip of a few bubbles come out here and there. So what we know is there's not enough bubbles to show up on the ship sonar system per se, but they're definitely there, which indicates a lot of gas charge underneath the seafloor sediments.
And the ROV has methane sensor?
That's right, yeah. Scott is operating the SAGE methane sensor, so it's measuring the concentration of dissolved methane in seawater. And we saw yesterday, as we went over those Seep sites, the concentrations would go up and then would go away from the site and they go down. So the sensor seems to be working quite well. We're today, in order to accomplish our mapping goals, we're about 25 meters off the seafloor. So we're going to be interested to see if any of that signal of the methane concentration makes it up to here to potentially be picked up by Scott's sensor as well.
And do you have an idea the concentration of methane there?
What do we have for the concentration of methane here?
So at the Seep yesterday, yeah. So it maxed out at about 170 ppm was our highest reading, but depending on where we moved around on the Seep, we were reading between 50 and 170 ppm. And then even at the seafloor, we're seeing elevated, only a few ppm, but elevated background methane.
Okay. To have an idea, the methane concentration in the atmosphere is about 2 ppm?
Yeah.
Yeah, 1.9 maybe?
Yeah.
Yeah. So it's quite high. in the seeps. So there are some microbes and also clams able to use this methane as a source of energy, as a energy source.
Yeah, that's right. There's microbes that can use the methane and the hydrogen sulfide gas that comes out of the seafloor as a source of energy, as the foundation of the food web for this ecosystem.
Yeah. Yesterday we saw also some worm tubes or tube worms. Tube worms that are sustained by these chemiosynthetic organisms. And also we saw, I think, microbial mats, several.
Yes, we saw a number of microbial mats throughout the area. They seem to be a little more to the periphery of the methane seep sites, as we saw them yesterday. And I was not here, but I understand at the end of the dive they found a bacterial mat that was sort of yellowish orange, which is a bit different than the typical white bacterial mat. So that's something interesting to think about.
Yeah.
What might be causing that?
Yeah.
Oh, that's a bump.
Oh, it's a bump on it. We're going over,
A bump bump.
we're Matt, well, I think, Adam, you drew the survey lines here. Was our first line going over the large mound?
Yes. No, this is not the large mound. So on the ROV map where you see all the yellow dots for waypoints, that's the large mound. We're a bit to the west of that, but this was a mound on the EK7 or EM712 survey.
Can you see on our M3 screen, we're going, I've got the color palette now kind of dialed in to where you can see we're going over that isolated kind of elongated mound.
Yep.
north south. So that's good. That's a good, mapping is looking good.
Methane is there.
Do you want to make it narrower?
No, until it's here. Okay. Tickle my nose the wrong way. No, it's fine. Yeah, so we'll be able to adjust the crops. Great. So you're in auto altitude. No, you're in auto depth. That's perfect. Perfect.
The only thing that might be an issue with that is the regional, the whole bathymetry here is sloping about almost 4 degrees to the west. So if we maintain a constant depth on each line, we'll slowly get away.
Yeah.
So our swath, it'll start getting deeper. So is it something that we want to, like, on each line, do like a reset of our altitude and
That's
then
easy
lock
enough,
it
To
in?
maintain
The-
25.
Yeah.
Yeah.
The survey lines are roughly contour lines, so that it should be constant depth along the line.
Yeah, that's just, let us know how much you
Yeah.
want to move, it's easy.
Okay.
Nothing hard.
I can push that button.
I can push that.
Squeeze.
Jeff, what kind of swath width are we getting?
Right now, I'm seeing about, we're getting soundings out to 35 and 35 on each side,
So almost
that's
70.
70, but I would say clean soundings are probably to about 30, 32.
Okay, so Our line spacing should be okay.
Yeah. It won't record the noise.
It won't mess with your color palette.
It won't mess with
Protect the color palette at all costs.
the color palette and my hands.
Yeah.
I want to have to clean this data.
Hey, did you look at the data that we recorded while sampling on bottom? You know how we were
The low altitude.
Yeah.
From dive one, no, I haven't.
I'm so interested to see
Like when they were 2 meters
if
off
it's
the
worth it. Yeah.
Put the other depth sensor in.
Yeah.
Sí, bueno, esa nieve, hay mucha nieve marina.
Fair enough.
We can.
Uy, un calamar parece. Sí, calamar.
Okay, yeah. Everything should be reverted back to INS.
Sí, muchos organismos. Y pececitos. Más nieve marina. Oh, era un tenóforo eso, ¿no?
Sí, era muy grande.
Muy grande, sí. La medusa. Sí, estamos mapeando, parece. Observando dónde está
Are you happy with having that camera there?
El
or does
metano.
it not show you what you need to see?
I'm fine with whatever. I can't see the float here.
Okay.
I
Do
can
you
s-
need it?
I can see if I get little shrimp caught in the filter, like
Okay.
yesterday.
If you want it,
No.
then I can leave it, but if you'd
It's just
rather
on
have
the
a
other side
different
of the
display, that's fine.
There's no other display that would be more or less useful for me. So
Perfect.
it's ideal.
We'll stick with what we have.
Thank you, Jules.
Yeah.
Oh,
It lets me see the random stuff happening starboard side of the sub.
Perfect.
Yeah.
Yeah.
eso, Marisa, ¿lo pudiste identificar?
Era un camarón.
Un camarón. Pero pasaron muchos gelatinosos, ¿no?
Sí.
Jellyfish.
¿Se han visto sifonóforos? Que son estos alargados que tienen un poco de bioluminiscencia. Pero como vamos igual a una velocidad relativamente rápida,
Rápido.
no se alcanzará a visualizar muy bien.
Sí. Ah, la medusa, ¿no?
Esta es
Grande,
la medusa
hermosa.
género Solmisus.
Son hermosas. Ahí hay otra.
Alright, that's the line.
All right, marking into blind.
Stop logging.
All right, that's it.
I was waiting and waiting.
Way left.
All right, moving to the next line.
Wow. Not to brag, but our depth sensors are working pretty good.
All right.
About time, right?
Bragging about stuff that should have
Brag.
been done yesterday.
So one of the
All right, I'm gonna start logging this turret line.
Roger that.
Logging.
You guys want me to face the line now? Right. No sweat.
No sweat.
Okay.
Sorry.
We're fanning.
Giving you a
Really
nice
covered
spiral
you
right?
up.
Sure. You call that coverage.
Yeah.
That's coverage that gets deleted.
I guess he's the.
Oh no, I use it a lot of times. They're soundless, they're soundless.
I could have had them move like this, and you just would have had so much right here. Yeah.
This is better. Thank you for that.
Oh. Okay.
I'll try to work with Alex Paris when things calm down. Regarding
so one of the organisms that we are looking for is Theoploca. It's a big bacteria, sulfur oxidizing, that was firstly described
End of line.
by a professor from Universidad de Concepción in Chile, Víctor Ariel Gallardo. So we
So when you
hopefully we will
turn,
find it.
You just do an evaluation if we're back at 25 meters altitude and then
We're
set.
roughly there, we're at about 25.3.
Okay.
We'll see if it does it. It doesn't stop.
Yeah.
That is.
It's not.
Yeah, it's going on. Right?
Yeah.
Okay. Alright, yeah. We'll abandon that line.
Quizás ahora que llegaron más espectadores,
Still on.
podría Lucía contarnos un poco lo que hicieron ayer con el grano.
Oh, okay.
Presentarse y además,
When they get online, I'll.
Ah, Lucia.
yes.
Here is Lucia. Lucia is oceanographer from Universidad de Chile in Santiago. So, Lucia, do you want to introduce yourself?
Ya, buen día a todos. Soy Lucía Villar. Estoy haciendo mi postdoctorado en la Universidad de Chile en el Departamento de Geofísica y estoy aquí a bordo del Falcor 2 intentando sacar muestras de, bueno, más que muestras, datos acerca de la temperatura de los fondos marinos en estas áreas de escape de gas metano. Bueno, ¿quieres que cuente la anécdota con los pescadores? Llegaron ahí unos pescadores intentando comunicarse con el capitán. Pues obviamente nadie les entendió. Tuve que hacer una especie de diplomacia entre el barco y los pescadores, comunicarles que estábamos acá haciendo una expedición, que tuvieran cuidado, que teníamos instrumentos bajo el agua, que tuvieran cuidado con respecto a no pasar por detrás de las líneas o que no pusieran sus redes de pesca ahí. Luego se acercaron nuevamente y en el fondo ellos solicitaban agua. estaban sin agua y tenían que pasar toda la noche pescando. Así que los abastecimos de agua rápidamente y se fueron.
Ready for the next line?
All right, let's do it.
Oh, it's a glass squid.
Strawberry squeeze.
Oh, strawberry squid?
Maybe.
Alright, we're on the move.
All right, we're logging.
Bridge, ROV.
So,
Yeah, ready to go.
Yeah, we're headed back north, and you can try 0.6 again. I bumped the ROV speed up on this one.
All right, 6, okay.
Thank
On
you.
with it.
Cheers.
Lucía, ¿qué largo tenían los testigos de sedimento donde pusiste los termómetros?
Los testigos de sedimento son de 6 metros. Sin embargo, nunca vienen llenos de sedimento. Penetran, no sé, 3, 4 metros, pero el mismo core, el sedimento que uno saca el testigo, solamente contiene alrededor de 1,80 m y el medio uno, y el otro alrededor de 1 metro. Así que nos fueron como mucha muestra para los geólogos. Yo pude medir la temperatura, sí, en uno de los cores, así que ya extraje esos datos, y ya los envié a Santiago para que sean procesados lo antes posible.
Y para tener una idea, ¿cuánto es la temperatura a 3 metros de profundidad del fondo?
Bueno, la temperatura va ascendiendo, de hecho, desde el fondo marino hasta el núcleo de la Tierra va ascendiendo, porque el núcleo de la Tierra es el que genera todo este calor, es la parte más caliente del planeta, y este núcleo va transmitiendo su calor a través de las distintas capas de la Tierra hasta que finalmente llega a la corteza. El fondo marino tiene alrededor en esta área 7 grados Celsius, y los fondos marinos, bueno, como son poquitos metros, está como también cerca de ese tipo de temperatura. Si nos fuéramos un poco más abajo ya veríamos los cambios grandes de temperatura, pero cuando nos acercamos a estas áreas de Seep, de emanación de metano, es porque se está filtrando líquidos, fluidos calientes, bueno, un poco más calientes desde el fondo marino. Y ahí veríamos una señal clara en los datos, digamos, de una anomalía positiva de este flujo de calor. Eso es lo que andamos buscando. El flujo de calor es cómo se transmite el calor desde el núcleo de la Tierra hacia la corteza. Ese es mi trabajo de investigación, de hecho. Y mi trabajo de investigación trata de buscar el flujo de calor de la Tierra, pero a través de los hidratos de gas. Y los hidratos de gas son una capa de hielo que está altamente concentrada en metano. Este metano es el mismo gas que tú ocupas para cocinar o para el calefón. Entonces es altamente solicitado por el planeta, digamos, es un recurso energético. Sin embargo, este gas se está viendo afectado ya que él puede sobrevivir, digamos, bajo los sedimentos de esta forma de hielo si tenemos la presión y la temperatura bien restringida. Si calentamos los fondos marinos, este hielo, digamos, comienza a derretirse literalmente y comienza a escapar este gas metano al medio marino. Suele suceder en algunos lugares como estos que se llaman seeps, escapes de metano, y al momento que este metano llega al suelo marino, digamos, toca el agua de fondo, comienza a oxidarse. Es muy raro que el metano llegue a la atmósfera. Sin embargo, acá en Chile tenemos un caso súper especial, que es la isla Mocha, en donde está saliendo tremendas burbujas de gas metano y están escapando a la atmósfera. Y el problema del gas metano en la atmósfera es que es 80 veces más potente que el dióxido de carbono a corto plazo, o sea, de aquí a 10 años. Entonces esa es como la motivación de mi escapando estos gases a través de visualizar los hidratos en unas líneas sísmicas que yo trabajo, que son como unas radiografías del fondo marino. Y una vez que identifico en la radiografía, digamos, el reflector del hidrato de gas, yo puedo ir siguiendo su trayectoria por el fondo marino y luego más o menos estimar por dónde está saliendo este gas. Así que esa es mi investigación. Espero pronto tener nuevos resultados para entregárselos a todos.
Super. So Isla Mota is in the southern central Chile. It's an island in front of Arauco Bay. You see? Yes. Yeah. And in the beach, there are that bubbles that Lucia mentioned, and bubbles of methane that directly escape to the atmosphere. So Mott Island will be a site that we will sample on the next days. So maybe we will have some pictures there.
Going over to the mound.
Yesterday also we saw some corals, no? Several colors of corals, and also a octopus, a red octopus, very nice. Now we have 50 micromolar of oxygen, so lower than at the beginning of this transmission. That is about 1 ml per liter. So we are almost in the oxygen minimum zone. Un siphonophorus. Look, it's beautiful. Tiene muchos. Maritza knows.
The difference between sprint, the pressure depth is which one?
No, so the left side is sprint depth and the right side is altitude.
So the oxygen is decreasing
It's
already.
There's some bonus for that.
And it's amazing that many species can resist these levels of oxygen. This is a ctenophoro.
And then in the data, you can be like the first 2 months. I don't think
So guys, did you see some manganese nodules? Nodules? No. Rocks in this expedition? No?
Not yet.
Not here?
No, we haven't. I don't believe we saw any manganese nodules. We did see carbonate rock on our
carbonate.
previous dives, which is very common around methane seeps. There's microbes that create the carbonate rock outcrops there. We did see some exposures of that in individual pieces of carbonate rock that had lots of organisms on them.
Mm-hmm. Too bad. Carbonate rocks. Bueno, ayer y hoy en la madrugada hemos estado con Montserrat muestreando toda la columna de agua con un equipo que se llama Rosetta, que tiene un CTD que les mencioné anteriormente, y estuvimos tomando muestras para medir gases de efecto invernadero en la columna de agua, nutrientes, y filtramos mucha agua para ver cuánto contenido de materia orgánica hay, esto que estamos viendo aquí flotando en el agua, para ver el contenido de clorofila, que nos dice cuántas microalgas hay, un proxy de la productividad primaria. Medimos amonio también. Así que hemos estado trabajando mucho caracterizando esta columna de agua desde el punto de vista biogeoquímico. Y con nuestra colega Ana Adamo también estamos sacando muestras para ver microplástico. See, something sad yesterday was the plastic in the bottom, some plastic bags. Covered by sand. And Maritza, what did you sample yesterday?
Ayer colectamos 3 tipos de corales, si no me equivoco. Un coral negro, una paragordia, que son estos corales chicles que uno los ve y es como si fueran de mentira porque de verdad parecen como chicle y por el color que tienen, que son rojizos. También encontramos corales de lignoides, que estos son como muy duros, en realidad son muy escamosos, y esa es la particularidad que tienen estos corales. También pudimos colectar un pez del género Coelorhynchus, lo más seguro es que sea Coelorhynchus chilensis, y este es el primer, o sea, creo que es uno de los registros más importantes porque de los peces que podemos encontrar acá solo hay registros de pesca de arrastre o cosas así, ya que salen como fauna acompañante y al llegar arriba llegan todos rotos, no llegan con las aletas, y bueno, esta es una buena instancia para tener imágenes de estos peces, tiburones, cualquier animal como vertebrado que llegan en malas condiciones normalmente por la pesca. Estamos sacando las mejores imágenes para poder tener registros de ellos en su hábitat natural.
Yes, that's super important.
End of line.
End of line. Copy that.
también estas muestras van a quedar en un museo.
Sifonóforo.
Uno se quede con las muestras para la casa. Estas siempre van a quedar a un museo como debería ser, con todos los permisos que uno debe sacar. En el caso de mis muestras llegan a la sala de colecciones biológicas de la Universidad Católica del Norte. Todo esto es bajo un permiso de subpesca, nada de lo colectado acá se puede hacer si uno no tiene estos permisos.
Next line.
Para que sepa el público también que es importante saber.
If you want on this line to start trying to get auto altitude bound,
Yeah.
this would be a good one for, it's just a crossing on my cross line.
And it's super important that it's the first time that this area is visited by a remote operated vehicle, a ROV, because as Maritza mentioned it, the species that we know about this site is from fishermen, but this is the first time that we have images and samples from an ROV.
Alright. We're at 25 meters.
Okay.
And El Quisco, the name of this site,
Can you make a
has
note on our next circle line?
marine protected area, so all this information will be used
On
to
altitude instead
to
of
the
depth.
managers
You can
of
do it
these
with
sites.
This line that we started with, but you can put in the next one for the
It will be
circle.
useful information. Yeah.
I have to get the ship set up too. You don't want the ship to come all the way down the engines.
It's up to you guys.
That is end of line.
The line.
Alright, let's just get ourselves shorted here. See us, honey?
Yes, we are mapping.
It didn't start going down. They wanted it to go down 25 or no plan, so I just dropped it on there. Manually. Just updated the speed on that one. Yeah. Okay. Maybe check on yours too. Making a turn.
Where's the bottom here?
We are 25 meters off.
25. So we are 25 meters above the bottom. Hmm. 20.
Alright. Ready for the next line?
Yep, let's do it.
We are on the move.
All right, we're logging. So we've changed this.
Yeah, another
survey
Medusa.
line
Great,
to
ROV.
be operated at constant
Okay.
altitude versus constant depth, which means the ROV will be attempting to maintain a constant altitude of 25 meters above the seafloor, which should give us a relatively constant mapping swath. The first couple lines are using constant depth to see if this is more stable, but it looks like all of our depth sensors are acting as they should, without any noise, so I think we should be able to maintain a constant altitude and get good data.
And Jeff, this is a new site. It's different from yesterday?
Yep, we are mapping the site we were at yesterday.
Okay.
Since the Multibeam, the M3 Multibeam had a cable issue. We weren't able to do this mapping yesterday. So now we're mapping the site we were at yesterday to have a high resolution map
Mhm.
of the Seep site that we did the dive from yesterday.
Yeah. So how long will we map? How many times?
Adam, how many survey lines do we have?
We have, looks like 7.5 survey lines and a
Yeah.
cross line. So 8 survey lines remaining, and we're gonna do mapping here for another hour.
I'm looking at our waypoints from yesterday on the ROV map, is that in our next survey line, we're gonna go over where they put a lot of waypoints.
That's right. That is the large mount that we saw at the beginning of yesterday's dive that had abundant chemosynthetic species. It's probably the largest mound we saw in terms of a carbonate mound there. And as we go over at 25 meters above the seafloor, we'll be looking at our methane sensor to see if any of that methane is making it up to 25 meters above the
Mhm.
seafloor. If we get a signal there.
7 minutes after we go.
Yes, we'll be learning that 7 minutes after we
Yeah.
pass over it. But
You'll switch
As
right
you
now.
can see on my screen here, we're doing,
yeah,
That's what it does with altitude.
and our
Yeah, we
I guess
just-
it needs like a longer filter, like a slower correction, I guess,
Which one switched to that?
let's switch it, yeah, let's go back to constant depth.
All right. That's constant depth.
Here, the pressure correction, this should remove that, but.
If we don't do pressure correction, it won't. That's
Right.
it.
Yeah,
Yeah.
but the more stable the platform, the more we can have some air wiggle in the pressure correction, and it'll still generate a pretty good figure.
Yeah.
Do you have any control over the speed at which it makes altitude corrections?
Estoy hasta las 8 aquí.
Like it'd be a slow filter?
No.
Don't correct it on like 1 second or 2 seconds, a 1 or 2 minute running average of altitude.
That would be some sort of flight parameter within range.
Okay.
I have no idea.
Yeah, it'd be kind of like your autopilot, whether you want to make heading corrections on every few seconds, or would you let it kind of drift?
Right. Yeah, that would be
But I think at this point, if we can, it'll be a better view for the screen, for the waterfall display at constant depth.
It does look nicer, right?
Can you make a note of that?
I noted that we changed the constant depth.
Okay. Bet that RV pilot might have an idea of currents.
Northish even now, because I'm wondering where, for 25 meters up from now,
Mm.
where is that plume going that I saw yesterday in the sensor?
It's true. Yeah, we know from sonar imagery of gas heaps where bubbles come out that the bubble plumes often don't go straight up, but will go to one direction or another, and will even kind of make these S-shaped sinuous curves as they encounter currents moving in different directions as they move from the seafloor up towards the surface.
But Adam, some of this methane dissolved in the water column, or just go up to the atmosphere?
That's a really good question. So in sites like this one that are relatively deep, 400 meters, what the current research tells us is that microorganisms in the ocean are consuming the methane, so that the methane does not make it up to the atmosphere. And they've done studies where they've taken ships and collected air samples above these sites, just over the surface of the ocean, to see if they could detect any methane coming out of the sea surface into the atmosphere. And they've found that for deeper sites like this, that doesn't happen. But we do know that there are shallower sites. So in the case where maybe you're in less than 100 meters of water, all the way up to the intertidal zone, like the Mocha Island seeps, that there is some amount of that methane coming out that makes it to our atmosphere. And of course, that's something we want to understand, because depending on how you calculate it, methane is quite potent in the atmosphere as an insulating greenhouse gas. So we're very interested in knowing if we have a source of methane making it to the atmosphere.
Yeah. Maybe it's the case of Isla Mocha seeps.
Yes, certainly La Mocha seeps. We know in the intertidal zone that gas is, the methane is completely going to the atmosphere.
And also methane is oxidized? No.
Yep. So that's,
But abiotic, I mean, with no organisms, with oxygen.
see, there are oxidizing reactions.
Oxidizing.
And yes, then you have methane dissolving into aqueous solution into the water itself as it moves upwards. In fact, deeper sites, there's a process by which a material called methane hydrate is thought to form kind of a shell around the gas bubbles to hold that gas in until they reach a shallower depth where and the gas can dissolve into the ocean.
Yeah.
See, there's been some work at seep sites, shallowish seep sites, where these surface waters have been enriched in methane. So you're moving methane enriched water to the sea surface, and then you get into the air-sea flux of methane. So it might not be coming out into the atmosphere bubble form, but you are moving methane to the shallow waters, to the sea surface, which is another way of exchanging gases with the air.
That's right. It's also important to note that although sometimes we don't, it may not seem obvious, seeps exist also in lakes as well as the ocean. And so there are shallow bodies of water, we know, where they're degassing methane, and of course that gas is likely increasing concentrations in the water and potentially getting to the atmosphere as well.
It's like, I think in wetlands also.
Yes.
The high amount of organic matter,
Right.
degradation, produce methane.
That's exactly right. Coastal wetlands are some of the larger sources of methane to the atmosphere on Earth.
We took samples to measure greenhouse gases, methane among them, in the water column. So we will compare between what's that? Siphonophora? It's another kind of siphonophora. We will compare a water column without seep and the water column on the seep. So maybe we can observe some changes in the methane concentration in the water column. So the oxygen here increased a little bit, but it's still very low. It's 18% of saturation.
This next survey line should be pretty interesting.
Yeah, here we go.
Going over all of the things we were at yesterday.
Good, maybe we'll sponsor the coral quiver.
When I mapped the coral quiver, I'll tell you. What's the size of the coral quiver? It's about a meter.
Yeah.
Yeah, so this is we're
Really
theoretically
taxing your skills,
at 25-meter altitudes with this many pings, we theoretically can achieve a half-meter grid size.
All right.
So that'd be 2 pixels.
All right.
So I'll take a look for 2 pixels sticking up. I think you know where it could be our geo-reference, because you know where the coral quiver is. If I find it, we can make sure the map is in the right geo-reference. Did you mark the other waypoint for the quiver?
See where the little red dot is right here. It's within that 5 meters.
Oh, is that a question mark that says?
Yeah, that was yesterday.
Oh.
That was yesterday when we thought it fell, and then it actually was over here somewhere, and then when we were actually leaving, it went walkabout again.
The coral quiver's just getting up and walking away?
Well, no, actually that basket decided that they don't want to stay attached anymore, so it just lingered down and kicked them out at some point. I'm like, wow, twice in one day.
You know, when it rains, it pours. When it rains, it pours quivers.
Right, yeah, unfortunately.
And then, Sonny will be back in a second.
Yeah.
Thanks, buddy.
Side? Okay, this is for Mike. So
Maritza, those medusas
stacked.
have bioluminescence. Did you see it?
Green.
Pero, ¿viste la que pasó? Tenía, ¿no?
No la vi, pero sí es una pequeña.
Sí,
I'm reading my notes.
en los tentáculos, sí.
Yeah, so side, this is all I have. Side
En verdad yo no sé
squares.
mucho de medusas.
We'll use the
Soy más
mission
de
requirement
fondos, de animales bentónicos.
driving
Pero
directly.
ahora lo único que
When blue.
Y muy bien.
we'll use the pilots specified. So that's that one. So,
Siphonov.
it's a good time.
End of line.
So
So if I
like,
said,
right now,
if I said this,
I would love starting.
I don't think you. So all it says is when green, engaged, we'll use the mission required heading driving from point A to B. So, I don't know, is that touchscreen?
yeah,
If you
And it's
engage
green.
that north, wait, which one's green?
yeah, that one does. That auto heading is
If you click
60,
go,
70 meters.
or actually, if
Then
you.
we have to set the heading in the waypoint, right?
I don't
So it
think
knows
so.
where to pull from.
I don't
How's it
think-
gonna know the turn? That's right.
It won't know unless we tell it.
It will go from A to B. That's all. Do you wanna just try it?
Only if you do fixed or a long line. Try it. Try and see.
Yep, alright.
What did you try?
We're gonna go to
Going
this little cross
the
line.
short one.
Yeah,
Yeah.
we're trying to sort out something on the software though, so.
Got it.
Alright, so go into the waypoint.
Okay, that's what we want.
Estamos girando, girando, girando.
No, but you have to have it fixed or draw a long line.
Ahí, ¿viste, Maritza?
Have it as-
That's a long line, 'cause I wanted it to turn. I don't
A
want it to
jellyfish
stay like this.
with bioluminescence.
That has to be set in order for it to take that command.
Yeah, yeah.
Okay.
Are we on this slide?
We are.
We're we're logging this one.
Yes, this is just going straight across
Got
to
it.
7 now.
With our little connector.
We're finding out little tips and tricks along the way.
Nice.
And then, so the
So
next
it's
one
gonna,
is
when we get onto
depth.
this next
When
survey
we
line,
use
reset the
mission
altitude.
required depth,
To, yeah.
if it's put in.
Yeah, it's like D3 right now. So we don't know.
There is another one.
We changed
I mean,
ours.
we have to change it, but that's not super useful.
Another.
No,
Actually,
that's
awesome.
altitude.
Right.
That's the problem.
Yeah.
The altitude is hunting.
Mm-hmm.
So we have to reset the altitude utilizing auto depth for that.
And then the bunny
Yeah,
speed,
right.
when green engaged, we'll use the mission required speed. Direct
Okay,
from
so
UI.
there. We'll set this for,
Can control, when
set this
blue,
for-
we'll use the pilot specified speed. Can control with the rabbit slider. Shay is disengaged now.
Okay, so
Okay,
you have
so
that option. You can just go to parameter.
If you have it
to
disengaged.
It's disengaged.
Hmm.
Yeah.
Bracket.
So do you normally change that?
No, because we're Doug
So
said
Adam, these are 40
It's just
meter
fine.
This is
swamp
perfect.
line spacing,
Mhm.
so
Oh,
that
38.
we've got good coverage.
Auto
I was
hold
just estimating. You
keeps
got
you in the
almost.
specified position that is selected on the navigation.
Ahí hay otro sifonóforo.
If the pilot
Sí.
is running
Oh,
an
because
auto mission, the pilot
your
can
line
enter
spacing is
controls
40,
into the joystick to
and we're
override all the
getting
navigation
60.
functions. Okay.
Yeah, so we're not 100, but
Ah,
no, I was just estimating.
Okay.
They also tried to teach me about derivative, integral, and proportional, but got 45% of it, and needed to do more research to be able to.
And the line.
In the line.
En la-
Yeah. So Pero's in, and it's solid, yeah.
Any
I
depth
don't
changes,
think
or we
we need
can?
to apply any
Return to
corrections.
25 meter altitude and lock in auto
Sprint
dash.
does
Okay.
the, yeah, I think so.
Moving up.
Either way, I didn't do anything, and it's.
So we're at, what it says, below the transducer is 23,
Yeah.
so we're pretty close, but still running auto depth.
Yeah, we can go, because
Yeah, I'm
it
sorry.
seems to slope down as we go north, correct?
Yeah, there's a deep spot in the northwest corner or the north part of the survey.
Alright, we'll come up.
Pacific Northwest.
PNW or?
That's where I'm putting it on. Or the northeast Pacific. Alright,
Alright.
we're logging.
Good. Should be
All right,
able to
start
go to the
logging.
quitting point. See if it does it again. When I set it. See if it turns the correct way.
Alright.
Making the turn?
Yep.
It's making the
Stop
turn.
logging, actually. I'll start when you get online.
Ahí estamos virando,
Let's
virando.
keep going,
Okay. It's online, and it's starting to head.
Okay, start logging.
yeah. Brad, you want me to update waypoint?
Starting to move.
Starting now.
It's cool, isn't it?
Yeah, that's what I used. Put that in where we're at, but-
I think we should get a copy of those notes.
Yeah.
Otra medusa.
If you want to after this long, you want to get breakfast. If you want to get breakfast after this long, and then you can go to the 9 o'clock breakfast at 30. Yeah.
Those headphones are good, man. You can hear them.
It's all this is protected.
It's just like really generating that metal.
Get pumped up.
Seguimos mapeando. Ya. Oh, tela. Ya. ¿Y aquí le voy a preguntar qué organismo es? Ya, seguimos mapeando. Bueno, este mapeo, como mencionaba Jeff, es súper importante para saber dónde están los ventiladeros de metano. Y es un mapeo de alta resolución que permite ver con mucho detalle lo que está ocurriendo en el fondo y si hay gas, metano o no. Y entonces poder enfocarse luego en estos sitios y ver los organismos, microorganismos, hacer muestreo de los organismos que están ahí, además rocas, sedimento, agua desde el fondo, y poder caracterizar estos sistemas y poder determinar con mayor precisión cuál es la importancia de ellos. How far from the boat is the ROV?
97 meters.
97.
Yeah. So that's the distance.
Ah,
This is the
okay.
boat. Yeah.
And the ROV can't stay in the red circle?
Yeah, so that's, the red circle is 50 meters from our launch point.
Okay.
So we always stay outside of that. Because the umbilical can get.
And the ROV can stay by itself 25 meters above them?
Yeah.
So it's connected by a cable,
Mm-hmm.
but he can stay at that depth?
Yes. Yeah, so we have it at that, since that's green there,
Mm-hmm.
it's an auto depth. So it's just keeping that depth.
Yeah.
So we can do altitude
So
as well.
he feel the bottom, or how it maintain 25 meters above
It's
the
using the pressure, right? Depth.
We're using depth, yes, but it also has a DVL altimeter. But yes, it's staying there with pressure.
And we will do all those lines with the ROV?
Yeah.
Not,
So you have
probably
to-
not all on this time. I don't think we're gonna finish on, when the next shift comes on, they'll probably wanna go do sampling.
So it's just set amount of times what we're doing. More or less.
For now, we
Okay.
could come back to surveying later or on a subsequent dive. We'll
Okay.
see what happens when Greg and Lisa, other people get out.
Start getting interested.
Yeah, we're already seeing a big mound. This
Oh, yeah, nice.
And what the people is seeing is that screen or that one?
The top left one.
The top, the small one.
Yeah.
Yeah, so we can
See some of the
see
sonar.
like in 3D. Rotate this In a different orientation.
That map also, they are seeing, oh,
So that green feature in 3 dimensions on the map is the mound that we saw at the beginning of the dive yesterday.
the 3
Those
squares.
at the end of the dive.
And at the end of the dive, they come back to it. Like it's,
Yeah.
very good. Oh wow, look at the sonar.
So that's
Yeah.
You see
Okay.
a quite tall
And
mound.
those are the chat comments.
okay.
And then
Do you think you're worth seeing those two
Okay.
structures on top, that double layer?
So when Jeff or Adam say something about this, that appear in the screen.
ooh,
Okay.
here it is.
Oh,
All right.
Very cool.
what is it?
Charts.
Marcela, lo que vemos acá es nieve marina, principalmente.
Sí, hay de todo. Hay nieve marina y organismos. Los que se mueven muy rápido son organismos.
Which where?
Off of the northern.
Yeah, it might be a little
right
high,
side
but
of the
it
shadow.
can't, it's a, yeah, the sonar shadow, you
Yeah.
can't see behind the pit. But yeah, that's cool.
Y sabes que es impresionante la cantidad de material particulado que hay. Si, bueno, el Falcor ha hecho varias expediciones hacia el oeste, en aguas oligotróficas, y, o sea que tienen muy poco nutriente y baja productividad, y las aguas son mucho más claras que lo que nosotros estamos viendo acá. Y eso yo creo que se debe a la alta productividad que tenemos en la zona.
De hecho, ese mismo carbono que de estos organismos que comienzan a caer en los sedimentos y se empiezan a acumular, son los que poco a poco se van degradando y también forman este gas metano.
Estos SIPs, sí. Bueno, estos SIPs se producen en zonas de alta productividad.
Put a marker that there's a small now.
Estos sí
Yeah,
son
I think that might have been waypoint 1 from yesterday.
That was way
de agua
yeah,
Sí.
probably not right.
Sí, los cold SIPs. Porque no tienen que ver con actividad hidrotermal.
That one pixel right there.
All right, that's it. Perfect. That's way far away though.
No, it's not.
Nearer to the
Y esos
mountain.
son mucho más
Mm.
profundos. Al parecer los que están en Isla Mocha, al sur de Isla Mocha, no en agua sonar, sino que más profunda, tenemos la sospecha que proviene de la disolución del
De los hidratos.
gas.
Ah, mira.
Por eso la emisión acá es como corroborar eso.
Determinar.
A través de los perfiles sísmicos.
Wreck the identification.
Y ahora quiero verlo en vivo.
En vivo.
That's all that is.
Bueno, antes de este sitio, que se llama el Quisco, visitamos Limarí, buscando un cold sip también. No lo encontramos, pero estuvimos muestreando el cañón del Limarí. Ese cañón está frente a Ovalle, en el inicio del norte de Chile. Inicio sur. Y luego de este sitio, el Quisco, nos vamos a ir a San Antonio. Al cañón de San Antonio. Sí.
So we got it dialed in right now.
Que está un poco más al sur,
Just for the ship.
muy cerca de acá.
It's all within the
Este es un gran cañón, ¿cierto?
Es el cañón del río Maipo.
Deja como una huella de digamos, la batimetría muy fuerte.
Ujú. Y ese río
and make sure it's all
uno de los ríos que alimenta el río Maipo es el río Mapocho, que cruza la ciudad de Santiago, que es la capital de Chile.
So that's a lot of just for
No, but I
take
get
a few more minutes. Stop on town.
Our baseline has been
Go to waypoint.
maybe a
Nine.
ppm higher
Up
over
to
the last
last
2
year.
years, but there's
Yeah.
enough noise and I can't read out the 10-second average that I'll hold off on saying anything.
Dudes turn
El
first.
tema de los ríos,
Start
cómo
far.
transportan sedimentos desde...
Turn dudes.
Mira, ese es un sifonóforo. Creo. Sí, son muchos. Sí.
Transportan los sedimentos desde la tierra
Keep it about 20
hacia el mar,
Seafloor.
y sobre todo en estos cañones
It's not
submarinos,
hooked up.
que son verdaderos ríos en el fondo del mar. Y este río, digamos, este río sí va transportando finalmente todo su material hasta las fosas, hasta las fosas de Chile. Y ocurre una cosa bien curiosa, que frente a Valparaíso, aquí cerca donde estamos, se encuentra, ustedes saben, la mayoría de los chilenos saben que está la isla Juan Fernández. Y la isla de Juan Fernández es un hotspot donde está saliendo lava, digamos, desde los fondos marinos. Y va creando esta serie de islas que forman un archipiélago, el archipiélago de Juan Fernández. Y debajo del archipiélago de Juan Fernández hay montes submarinos de origen volcánico. Y estos montes submarinos se van moviendo por la placa de Nazca hacia la sudamericana, chocan con la sudamericana, se subducen. Y al subducirse, justo ahí, como tienen mayor buoyantez, estos montes submarinos como que levantan un poco la placa en ese punto y hace una especie de barrera, una barrera natural de los sedimentos. Entonces, si ustedes ven un mapa batimétrico o en el Google Maps, ponen la parte de satélite, ustedes pueden darse cuenta que de Valparaíso hacia el norte el sedimento es muy escaso en la fosa marina. Por eso en Atacama tenemos la segunda fosa más grande del mundo, casi 7.000, 8.000 metros de profundidad. Y obviamente también porque no tiene aporte de río, ahí es desierto. Y de Valparaíso hacia el sur tenemos la fosa súper llena, rellena de sedimentos. Así que es un dato como bien curioso geomorfológicamente, la dorsal de Juan Fernández.
So we are in the subduction, where the Nazca plate goes down to the Sudamericana plates, no?
Yeah.
So we are part of the
When we start the next one, or even on this,
hot ring,
the next line, just double check
no,
the
hot Pacific ring? I don't know, how can I say it?
Ring of fire.
Ring of fire. So
Downhill on each line.
Yeah, we'll just resend it.
that's the reason why we have several earthquakes, big earthquakes. But also is the reason because we have the Atacama Trench. We are mapping for, I don't know, maybe 12 more hours. No. And after that, probably we will go to the bottom, to the seafloor to see.
Deep sea research podcast.
Yeah.
Don't forget to like and subscribe. And ring the bell. If you want to keep getting notifications.
Actually, in the Ring of Fire, we can find also all the volcanoes, active volcanoes in the world. Most of them are there in the Ring of Fire, which cross from South America to Central America to North America, then
All America.
Alaska, and goes to Japan to the west, and then goes down until Indonesia, I think. So it's a big ring. and then you can find the major earthquakes, the super volcanoes also, very active volcanoes. Our active volcanoes is called Villarrica volcano actually. And we have, the earthquake also in Japan.
Okay, so you're not, you don't.
The earthquake, for example, and not only the earthquake. Do you remember the Hunga Tonga Unga Hapai in
Yeah.
Tonga? That
Oh, you did? Yeah. Oh, magic. Alright, it's come down to 25 meters.
We have also some volcanoes explosion, no?
yeah.
In the southern Chile.
Yeah, actually, the last cruise from here, from Falcor 2, they were looking for lava. I mean, debris and lava sediments in the from the Chaitén explosion.
In the fjords,
Yeah,
in the Patagonian fjords.
Mhm.
No, are you good next? Sorry. Yeah. No, we're not doing well. I guess are you good next? I'm doing a right click up there. Yeah.
Siphonóforo
Yeah.
and Medusa, jellyfish.
Oh, okay. But you could potentially set everything up and then just sit back and back. The only thing is they want to keep it about 25 meters. So just talk to Jeff if he wants to come down. But yeah, you want to come down basically start 25 meters every line.
Yeah, every line kind of reset back to 25 meters and auto depth for the whole line.
Cool. Yeah, so you're in depth right now. Yeah. So we're in depth right now. So you would be.
So there is an active carbon pump here. A lot of marine snow and many organisms at this depth. 389 meters.
Just whatever happening, I'm going to probably be like, all right, let's do it.
Yeah. So I'll show you the way to make it and do the next one. Make it so you can sit next.
Marcela, do you know if the reason for this explosion of life here is because of upwelling?
It's the upwelling. It's a consequence of the upwelling. Here we have winds blowing from the south to the north, to the equator.
When you're straight on line, you ready? You're good? All right, let's start.
So, because of the Earth rotation, you have a movement of the water, the surface water, offshore. So this, a kind of space created by this movement is filled by subsurface waters rich in nutrients, and also cold waters and with low oxygen, such as we are seeing here. So these high nutrients fertilize the surface ocean, and the phytoplankton is blowing, and as they are the base of the trophic chain, all the trophic chain is enhanced by this process, the upwelling events. So for this reason, the sediments are very rich in organic matter. So as you mentioned before, the decomposition, the degradation of this organic matter generates methane.
That natural process is called methanogenesis.
Methanogenesis, yes. This process is occurring when no oxygen is available.
That's a long.
So these animals and this plankton that we can see in the screen, they depend on the sunlight, right?
Not directly, but yes, because they are part of the trophic chain that begins with the phytoplankton that needs light to do photosynthesis.
Okay. It is true that I hear sometimes that ocean give us the 50% of the oxygen in the atmosphere. Every two breaths, one breath.
In fact,
Comes
I
from the
think
ocean.
it has been estimated that it's between 50 and 80%.
From the ocean.
Yes, from the ocean.
So we have to be
Yeah.
careful with the ocean.
And there is a tiny, tiny phytoplankton named Prochlorococcus that lives in the with penumbra. I don't know.
In the shadows?
In the shadows, but they need a very little amount of light. So sometimes we can see it as a secondary maximum in chlorophyll, and they produce about 20% of the oxygen in the ocean. They are very tiny, but they are very important for us.
Yeah.
Yes. What do you need? Okay.
The seafloor is getting more reflected right below us. This is just more of the extent of the large mound that we were sampling at yesterday. SAGE in this group. Up. So there's some enriched methane over the mound, even at this altitude. Once the geotip is loaded, is the color scale set. Can dynamically change the
Twilight.
range. Okay.
Of what? The color?
In green sea,
Not on green
the
sea.
geotip.
It's just, yeah, but I'll adjust the enemy proc.
this is what after the survey's done, I was gonna go try to make a quick map of the M3 data.
Okay.
But I can put the geotip into the
Yeah.
dive folder.
Yeah. And then we can bring it into green sea.
RV Bridge, would you like me to make a move?
Just kind of saying
Vale,
what
vale.
they've been doing. I would do that full move.
Otra medusa pequeña.
The line that I'm on right now. No, no, like down, see how they kind of straight down, like how they're doing that zigzag. That's probably about as far as we need to go. So come down that way. Right there.
¿Ese era un tenóforo?
Sí. Una de las características de los tenóforos que tienen estas banditas bioluminiscentes es como...
Es como un globito, ¿no?
Sí.
Y que tiene bandas de bioluminiscencia.
Y algunos tienen estos como tentáculos, dos tentáculos en sus costados.
Start the move.
Este tenía.
Sí. Hay otros que no tienen o los tienen muy cortitos.
Oh, ¿viste?
No estaba notando al cámara.
Es un tenóforo con mucha bioluminiscencia. Muy hermoso. Hay muchos organismos gelatinosos. So Jeff, for the new audience, we saw methane now or not yet?
So methane, we haven't seen any bubbles. We haven't seen any bubbles coming out of the seafloor in the acoustic, in the multi-beam mapping.
Mm-hmm.
But we have gone over the large mounds that we were sampling at yesterday. And those seem to be semicircular and isolated.
Along with a few spikes in methane.
And yeah, and SAGE from across the room seems to be showing some elevated methane as we've gone over the center of our survey, which corresponds with our Seep that we were diving on yesterday. So at 25 meters altitude, SAGE is even still picking up a enriched methane concentration in the water.
So how can we see in the picture in the screen if we find a methane rich environment? You see like a red line?
So in the color screen, I keep having to change the color scale because the depths are changing. But when I have the color scale tuned correctly, you have these circular high spots, and the color is depth. So the reds and oranges are shallow, and the blues and purples are deep. And when you see isolated reds and oranges or surrounded by blues, that probably means it's either a slightly elevated mound or maybe a small carbonate outcrop. The Multibeam map is not a direct measurement of methane, but it measures the structures that the methane seeps produce. So the way the methane seeps alter the seafloor, we can map the seafloor and see those changes, which then, when we have the high resolution map, it can instruct us for where to go with the ROV down on the seafloor, for things that we might have missed if we were just on the seafloor looking around with our narrow field of view. So this map hopefully will aid us in finding maybe some spots that we wouldn't have found if we were just searching on the bottom.
And what area are we covering now?
So we're surveying about a 60-meter swath. So you can think we're making a 60-meter by however long the lines are, 250 meters long by 60 meters wide swath of coverage, where if we see something in that, we'll be able to go back and look at it. We're mapping right now just southwest of where the big tube worms and seep was yesterday, when we were diving on the and doing the sampling. And what I'll do after we finish our survey is go in and process the M3 multibeam data and stitch together all of our survey lines into, I hope, a really cool map of our site with a level of detail that would be the size of the ROV. Essentially, we could find things smaller than the ROV on the seafloor. In addition, as we talked about before with SAGE, doing this survey in a grid pattern, SAGE gets to get a really good data set of how the methane concentration increases around the main seep site in a consistent way. So he'll be able to grid this maybe into a heat map of methane around our survey site.
That's it, end of line.
Okay, end of line, stop logging.
Moving over.
So most of our work does not end here. So
No.
it's the beginning
This is
of
just the
several
beginning.
years of work.
Can we get to the next slide? Did you have a grid tree? I'm gonna log the turn, or
You wanna
part of
log?
the, I'm logging part of the turn, but you can keep doing what you're doing.
Okay, do you want me to do when we're starting and stopping the short end? Is there just
It
a-
helps.
Okay.
Most of that data is not gonna get used, but we are looking outside of our survey box, and if something's out there, we'll see.
Well, I'll let you know when we start and
Yeah.
stop this.
Okay. So if I clean up the Emmy Brock. Thank you. Yeah.
stop logging,
Just coming down to 25.
adjusting. There's something, maybe a squid. Kind of see it in the sonar. Can you get to log name 3? I can have Adam do it.
I can.
Okay.
Did you eat?
I'm about to, that's what I was
Oh my
going to get
gosh,
to the
yes, absolutely.
What should I do? There's some good
You
stuff.
should get the chia, black forest pudding.
And a churro.
And the
Starting the
churro.
lab.
The churro? My goodness.
Starting the light?
All right, great, thank you. Starting logging.
So you're not logging the turn data?
I'm trying to not do the rotation
Yeah.
and then get as much of the straight bit,
Okay.
but it's not super important.
Yeah.
And then I've been kind of just adjusting the palette to make the visual looks good because it.
Let's see. Probably start
You'll
looking
see
up
it.
right as we.
And then this is probably wide enough, the two depth cables,
Yeah.
but for
Mhm.
some reason, if it's starting to get really shallow, you can slide the min depth.
Yeah. Awesome. Got it.
Thank you.
Enjoy.
Beam, beam. So we still 25 meters above the bottom. Oh,
Oh,
that's
that's what was in the M3.
Oh.
Yeah, I think
Yeah.
we saw that guy on sonar.
Yeah.
Él quiere que lo veamos. Otros.
Do that.
Oh, full.
Oh, there's mound. Hard reflective. This is probably what we saw in the last line now. Oh, no.
No, this is
Other side.
another mountain. This is
Sorry,
waypoint
yeah,
2.
Yeah, cool.
Yeah.
Yeah.
This is one of the two mounds that didn't have any methane with it yesterday.
Hmm. Interesting. Okay.
You can see it there.
Esta esa medusa con
Wow, you just got
tentáculos
Double it.
bioluminescentes.
That was a lot of ink.
We're seeing another mound in the
Yeah.
sonar again. This is great. This is going to make a really nice map in this area.
Yeah.
Complement all the ROV explorations going on and give some really good context, spatial context to what we're seeing.
Bridge RV, you can move to the waypoint.
Are we hitting the bottom depth gate?
No, it's
No.
just the
Drop it.
display.
I got
Like kind of advance them forward. They're not going to be running away from us when we go across. Or whatever you want. I guess that might work. So you're thinking we'll come straight up and then.
Yeah.
I understand.
as I'm looking.
The sonar resolution is probably just a little too low to pick it up. It would be a few pixels on the sonar, so
Yeah.
in theory you could see it, but it'd be tough.
Yeah. But
We'll definitely look for it.
we'll look for it.
For sure.
Yeah.
It would be a few pixels with a relief of a third of a meter.
Yeah. Which is within the envelope of resolution for the sonar, but it would be
Certainly.
it'd be very, very small objects.
Just thinking we have a pretty big, decently large depth range on our color
When you
board.
get to the next line,
Yep.
can we set our other depths?
I did last time.
Okay, perfect.
Yeah, it's just going down.
No, yeah, of course.
Right, so did you
It'll
want to
come
do it
back
at the
up.
shorelines? Because I was doing it before the long line.
Yes, no, that's
Or
perfect.
do you want me to do both?
No, just the long line.
ones,
On our map over there, we're just going into a divot now, but it's fine, we're within the range.
What's the range, like what's the high end that you want to see? Like, you start seeing after a certain
Mm.
number?
I don't know if we do it mid-line, because that's not what I was doing. I wouldn't do it mid-line, but I just need
What's
to
the
make sure I
size
have my
of the
range
pixel?
gates fine.
What's that?
Somebody
Otherwise,
is
it'll just
asking
ignore
the
it.
size of the pixel.
Yeah.
Oh, yes, we have a couple questions about the sonar.
Yeah.
The swath of the sonar system we're using right now in our ROV Sebastian is approximately 60 meters across from side to side. So we're mapping a stripe of the seafloor that's approximately 60 meters wide. And each of these pixels is going to be around a meter square, perhaps slightly smaller.
And so we're still mapping yesterday's dive there.
Yeah, mapping. It's a great map.
¿Qué toñato? Tenóforo, tenóforo. Tenóforo, perdón.
Today's so they're not using the same terminology. It's going to be very confusing. So the big spike was over the giant where bushes slowly went over. How much longer you guys just estimate?
Until handoff, we can break away from the line.
Well, but it's almost that time that if you want to finish.
We'll,
I mean, if you're within an hour of finishing, you probably should.
okay. Yeah, it'd probably be another hour to finish.
That's good.
Okay.
I think that's better than
So
requiring
on your
you to come
screen,
back here
we're
if you
displaying
wanna dive
both
somewhere else.
the
Yeah.
4K camera,
Yeah.
but we're also showing you live output from our M3 sonar that's currently on ROV Sebastian. On the bottom right is the water column view, and so that's all of the active returns coming from the sonar. So if you look very closely, there's a bunch of green squares throughout this assumed bottom, and that's the strongest return the multi-beam is receiving. So when the beams get sent from the multi-beam, they go until they hit something and return back. And so the green dots are the multi-beam's assumed bottom due to the strength of the return. And so they're dancing a little bit, and that's just because the bottom isn't exactly flat. And so those returns then get computed into the top display there, which we call the waterfall view. And your view is if you're laying on the top of Sebastian looking down towards the seafloor. The scale is so that purple is deeper and blue, green, headed up to red is shallower. So on our east side of the vehicle, it's shallower and it gets deeper to the west. you might see a bunch of rainbow colors on the left side, and that's noise. That is data that will have to be, those points will have to be edited out in post. And that is from the multibeam picking up some stronger returns towards the edge of our swath. And so that's quite common in multibeams, is you have a noisier edge of the swath. And so if you look at the bottom plot, you'll see green dots slowly appear, and that's the noise that gets put into the waterfall view. Okay. So Soph just displayed another map, and that's our MB Proc 5 computer, which is displaying the ship data that we, actually this data was previously mapped, yeah? Or is this ours?
This is ours from the 712 survey we did on the first day we were here.
Can I steal that computer mouse, please?
So the MB Proc is the 712 data, which is our shallow to mid water multibeam. And so we're monitoring this just so we can apply appropriate settings to the M3. Same with the MB Proc, or same with the M3 color scale, is purple is deeper, and red and oranges is shallower. And so we know that we just got out of a deep pocket, and so we're almost to waypoint 12, which means we have one more line to go to the south from waypoint 13 to 14, and that will complete our M3 survey. I'm gonna need the mouse in a minute.
Yeah. There you go.
Thank you. We have quite noisy outer beams. I'm wondering if that's a interference from another one of our depth sensors.
We saw that the earlier lines in the survey. Yeah,
Yeah.
it could be interference, but where it is, it's going to be relatively easy to clean.
Right.
Just strip it out, and as you said, in post-processing.
And why do you have these interferences?
And the line?
Huh?
The interference.
Okay.
Yeah, we have interference.
And a line.
That can be caused by a few things, where the sonar itself is using sound to measure the seafloor, so it produces sound that travels and reflects off seafloor, as Juliana explained. And so if there are other sources of sound in the water, that could be
Roger.
other sonars we're using, for instance, to communicate with the ROV, or to determine its position, or it could be even sound produced by the ROV itself, just the motors turning and hydraulics running in it, that can sometimes produce a little bit of interference, and that's to be expected. And as I said, we have a process by which we take these raw data and clean them and can remove those things that we clearly recognize as interference that don't represent what the seafloor is.
And there is a question in the chat. They ask about if the organisms can have some kind of signal.
I'm not. I think certainly we imagine that sound in the water is going to be perceived by organisms, and they may or may not react to it. But in terms of influencing their behavior, causing them to return or move away, we don't typically see any indications of that. So for instance, when we turn on the sonar, we don't see changes in the behavior of organisms around us that we might perceive as them reacting to the sound.
But oftentimes, like we noted before, we're pretty sure that we were getting some erroneous points in the multibeam during a few lines ago. We're pretty sure that was a squid that probably was below us, that the multibeam had some strong hits on it, and it assumed that was the bottom, when in reality it's just noise per se in the water column and the line.
Yeah, that's exactly right. And I mentioned sometimes sources of sound in the water can cause interference, and there are occasions where we see in multibeam sonar marine mammal vocalizations, like whale calls will show up as interference briefly in our mapping sonar.
And also the marine beams is used for to study the migration, no? Some faucets.
Like the diel deep scattering layer migration?
Deep scattering.
Yeah. So the sonar can pick up the plankton in the water column, what we call the deep scattering layer, and each day the plankton will migrate from deeper in the water up to the surface at nighttime and down lower during the day. It's actually considered to be one of the larger migrations that happens on Earth. It happens twice a day, every day, as the organisms move up and down with the light and darkness. And you can detect that with sound and with the sonar system and measure that behavior. One of kind of the cool little stories I heard about that was there was a ship that was surveying during a solar eclipse, and the deep scattering layer organisms responded to the eclipse. They actually migrated upwards a little bit as it got dark from the eclipse, and then when the sun came back out, they went back down. And so you see this little blip where they come up and go back down during the eclipse that's separate from their normal daily migration.
I think that they also respond to the moon cycle. So when you have a full moon, they go down deeper.
Oh, that's very interesting. Yeah.
We're a little far over.
Where's your RV?
Everything's perfect.
View waypoint. That's great.
Hi. Actually, we were getting
They're
a
gonna keep doing the survey, so I'm wondering, do we need
that. I'm wondering if that was
Where's
squid.
your RV? View waypoint.
me? You can go.
Oh,
Right.
thank you.
No,
No, I need to measure.
Yeah, that's fine.
Are you talking? Are you able to?
Yeah, I think
Hi.
Yeah.
Hi.
I mean, Jeff also.
Here.
We're gonna have you continue surveying. till you
So
finish.
right now
Great,
there's
we're-
kind of
Wanna
this
sit here?
glowing object.
I mean, I
It
can,
disappeared, but
but
a
I
few
have
seconds
so much
earlier there
rather
was
Oh, yeah, you
continuous
that's fine.
returns from an
40
object that was
minutes,
moving.
so-
That was
Okay, yeah, no.
probably a
We
squid.
have
one line.
Same with
And
here.
then a hash, and then a tie line, and then that's the survey. So
In the
everything's
M3
taken
data.
about
Like
30
20 minutes.
20 minutes a
20.
line, so 20, 15, so maybe another hour.
He said 40 minutes 10 minutes ago.
That's
so-
a rough estimate.
Okay. It's okay. Okay, so yeah, I'll
Can
see.
we try to be done by 9? Yeah.
I think that's gonna be close. Yeah, it should be about that.
I think it just makes sense.
Yeah, we're almost done. Great, yeah, I'll sit down,
We'll sit here and talk. So you guys want a logger? Probably. I don't know where Aaron is.
Yeah. Yeah, for any organisms in the water column.
There's literally no way.
Can you get back?
I'll keep running it.
Keep running it?
no worries.
Yeah, I mean, we can have you in there straight off also.
All right, hello again. This is Jeff Beeson from Oregon State University. And I'm back in the lead seat to finish up this mapping. Got Adam in the back.
Yep.
And we're cruising along. The ROV is moving around 0.3 to 0.4 meters per second on a bearing of 180. So we're heading south on our second to last survey line. And then we will do a tie line to, as you might have guessed, tie all the survey lines together. And then we will have completed our M3 high-res multi-beam mapping of our study area. I'll be handing over the ROV to our biologist colleagues to go down and do some more sampling and do some more video observations of the Seep sites. This was a pretty cool area yesterday, and I'm glad that we got the M3 fixed and this survey done.
So how long is this going to take you to survey, do you think? Like rough?
How long is this going to take me to process?
Yeah.
It's possible if the sprint depth is good, which I think it is, and the data has looked relatively clean, that should be able to get a map in maybe 30, 45 minutes of a rough cut. And then I could spend a few hours ping editing all the outliers, noisy data. And that would probably generate a pretty good one. And then I can use the cross line to look for any offsets. So when the ROV is flying, you have all your reference of the sonar to the reference point, and you know how it's. Oh wow,
They
there's a
should go
cool
there.
mound.
That is a mound to visit. Yeah.
We should go there.
Let's drop
You
a
see
waypoint
that?
on that.
Can we drop a waypoint? Is this? Oh yeah, we're kind of outside. This is a good
Yeah,
spot.
this would be a new spot. Yeah.
They all fall right along the center line of this depression.
That's because you have good mappers.
We have very good mappers.
Providing good lines.
Oh no, the center line of the depression in the bathymetry.
Oh, I see.
Yeah.
But also our mappers.
The
are
mappers
wonderful.
put
that.
The mappers put that there.
Yeah.
So Jeff buried a tank of methane under a mound.
The permitting was quite extensive to get that done. Yeah, so the cross line where we go over at a different direction, we can see any other offsets or biases that the ROV has got in the bathymetry. So if it's kind of leaning to one side or pitching forward a little bit and it's a constant offset, we can find that and correct for that as well. It'll be interesting. We went almost right over that. If SAGE gives a little bump on that one, because that seemed like
So,
really punctuated.
West of the big mound, I've had an elevated background compared to east of it this whole time. I'm
So-
wondering if it's related to that.
What can our ROV pilots? Do you have a sense of the bottom currents here? Not much, if any. So if there's even a slight current, any enriched methane would come up and move with the current. So maybe we're getting on the other side of the mound, facing it,
Or we're seeing that big one.
or
it's
it's just a
going
more
east.
active
Yeah.
spot. Yeah.
But if you look from there,
From our OS38, it's showing almost no current, but the current that is there is going to the southeast.
well, you can, if you.
Okay.
Oh, okay.
Yeah.
That's
Lower.
southeast.
And that might.
No current. That's
Which makes sense.
There's almost no current.
I saw an elevated baseline on the survey line southeast of the big mound.
Oh.
I saw
Well,
my
that's great.
first
That's
elevated
when you-
methane was
yeah.
on that one. So, yeah.
It's nice when independent measurements agree with each, kind of
Yeah,
line up.
and they tell you the same story.
To tell the same story, exactly.
Yeah. So Scott, what were you able to look any more at the SAGE data from yesterday's dive? I saw your initial plots looked. Did you make any initial plots from the SAGE?
Yeah. So there are a couple of put together a couple of maps. The big spikes were all laying right on top of that big mound that we were at.
Yeah.
And then the baseline. Now I have to look back at them, but the baseline along our transit from the east of that was a little bit elevated. So
Wow.
something tells me that might help us indicate that current. Look at that thing.
Yeah, I think we need to let the folks who come in the next shift know that that's a really really enticing target for ROV exploration of the seafloor today.
Yeah. So Yep.
Got a waypoint dropped for
Got
them.
a waypoint. If they need to, they want to keep going and looking around. That was very circular, and with a little peak in the middle,
Almost like a little,
almost like a pimple. Oh, we got a squid. Another mound? Yeah, it looks like we're getting another little mound. I was thinking, we were doing the constant depth, coming up on another little mound or high spot. That probably, I mean, it's making the M3 data look good, but it's also probably better for SAGE data to be doing a constant depth instead of constant altitude, where you'd be maybe moving into different water masses the whole survey. So another added benefit of going constant depth for you.
The depth could be, 10 meters lower. It would be even better for SAGE, but...
Hey.
Hey now.
I respect the M3.
Thank you for acknowledging that.
Well, and that speaks to the interdisciplinary nature of this work. We have scientists who are interested in biology and getting imagery of the seafloor. We have scientists who are interested in mapping, interested in deploying instruments, and so we're constantly working together to figure out how to combine those and compromise between the sort of requirements of those different types of data collection.
Jeff, I know we don't have a sub-bottom profiler on right now, but can the brightness of these returns tell us anything about not the subsurface, but the surface itself?
Yes. So we're
Softball.
look at that squid.
Really glad you like that question.
Yeah. So the multibeam is collecting it. What take a step back. So what you get with the multi-beam is detections. You're detecting the seafloor at different beams. And in addition to the detection, the time it takes for the sound to get to the seafloor, you detect it and it comes back. That range can be converted using the sound velocity into a depth. You also get an amplitude of the return. The detection has an intensity. So we can take that intensity value and also grid it and look at how the seafloor reflectivity changes. And that change in reflectivity can give us insights into the character, physical makeup, and the roughness of the seafloor. So yeah, we can use that as another product from the multibeam, and it can give us indications of small changes in the substrate. Like, is it clay? Is it sand? Is it rock? Is it carbonate? Is it rough seafloor versus smooth seafloor? So when you're seeing out there the line of dots, that's actually colored by intensity. So if you see it go to red to like yellow, that means it's harder.
And that's helpful for us in identifying potential seep areas, because we know that in areas like this, the seafloor is typically sedimented, which is relatively soft, but it's authogenic carbonate rock, and so that harder reflection there helps us pick out potential seeps.
End of line.
End of line. Start of line.
So in other expeditions, we've done this type of high-resolution mapping using an autonomous underwater vessel, an AUV, to not take away very valuable ROV dive time by, as you may have noticed, we're not seeing the seafloor. We're doing these survey lines at 25 meters altitude, which we do run into some animals, things that live in the water column, but we don't get to have that great view of the seafloor. Another expedition is using an AUV, but that can come down ahead of us and do mapping and generate the high resolution images of the seafloor ahead of time, and then we plan our dives using that map. But since we didn't have an AUV on this cruise, utilizing the M3 mounted on Sebastian gives us the ability to create these maps without needing an AUV and have a little bit of trade-off of ROV time on the bottom, which ultimately ends up being being valuable enough to justify the extra time needed to map. Start of line.
The screen grabs for these are going to be really, really good. Yeah.
In between, yeah, so it's better because of
Okay, we've started our final survey line. This will, based on our shipboard mapping, this is the rough extent. I bet we could find more if we kept heading west, but alas, there's only so much time. That we'll come up here to halfway through our normal line lengths and then turn east into a crossing line between all the survey lines we just completed and call it good, and then I will sign off and go on the computer for a few hours and try to piece all this data together. Hopefully by the couple hours or maybe an hour, if things are clean enough, that I can get a map onto the under the ROV navigation screen. So we'll put our glasses on. We'll go from fuzzy to clear of what the seafloor looks
That's
like
right.
around here.
That's right.
And if things go as I expect, what we're essentially changing is about a 10 meter grid. So a pixel being 10 meters by 10 meters, we'll go down to every pixel being 1 meter. So a 10 times increase in ideally resolvability.
Yeah, I think really good analogy for that is if you imagine you have a flashlight and you're pointing at the ground and you hold it pretty high off the ground, it's going to illuminate a large area, but you may not be able to see it too clearly because the light's farther away. But if you move that flashlight really close to the ground, you see a much more detailed, you can see in much more detail, but you only light up much smaller area up. And that's sort of the trade-off here. We can get close to the seafloor with the ROV and see things in a lot of detail, but can cover far less area than we can with the ship, which is sort of like holding that flashlight really high off the ground. So it comes down to sort of nesting these different surveys to best serve the needs of science.
Mm-hmm. Yeah, we've been holding the flashlight pretty close to the seafloor the last few hours.
Yes, this is high detail.
Oh, it looks like we've got another little mounds on the multibeam screen coming up. Now, it would be really cool if we saw a bubble stream flare coming out of the seafloor, which we would also see in the multi-beam screen, the bottom one, the kind of upside down ice cream cone looking display. Maybe you would see a high amplitude, a bright spot, kind of narrow and either vertical or somewhat laid over due to the currents. And that would give us a pretty good indication that there's a bubble emission site on the seafloor. But we have not seen that so far here. It looks like most of the gas has been trapped in the pore space and dissolved in the water column. Dissolved in the water. So SAGE showing just in general we're elevated now that we're on the west side.
Yeah.
What's our background here?
It's a few ppms higher than before. So
Cool.
if we set water column to 0, it's probably at about 4 or 5 ppm.
So we're completing our 8th survey line and 1 crossing line. We're about to finish our last survey line and then 1 crossing line. And these were 250 meters long, and they took, did you say 20 minutes to do a line?
I think we had 25.
25 minutes to do a line.
And this cross line should have quite a lot of features.
Yes, so we're good. This was a smart move by Adam, who designed this. Good to have a cross line over all the fun stuff. So we end on a good note.
And
yeah.
we get to image it twice, so
Yeah,
that
so
can
good
help
view
give
of-
us a better map over the really interesting areas.
And I guess that SAGE might like it also.
I expect that would happen. We'll see.
We'll see.
Or based on the trend, we'll actually just go down as we go from west to east.
Right.
Yeah, that's
So
right.
what does that mean? We're missing something huge west?
No,
I think we just flew over the huge thing in the
Oh, yeah,
west. Yeah,
that
that
mountain.
huge mound we saw.
Yeah.
Yeah.
It was very,
Do you know where the line's gonna end?
what's that?
it was very pointy structure.
Yeah, there's a pinnacle, and that could be the large tube worm cluster
Be far away from.
living on the peak, where that might indicate that's where the most methane is coming out.
Yeah, we're about to go over it again.
We're gonna turn and go over it again.
It's very large amount of the pinnacle. So if you get a chance, I would suggest we put a waypoint.
Yeah, but
down.
I gotta go to that orange bacterial.
Of course,
Everybody scores first, probably.
Just a suggestion. Yeah,
Okay.
absolutely.
So
Okay,
are these
These are the waypoints you
Greg's
provided.
points?
In
So
Greg
I
and
need
Viola
to
and-
figure out which one's the orange. You have a copy of the plan?
Yep.
Waypoint one. Over here. This is not.
all right, we're turning.
This was the mound we were at yesterday. This one looks larger.
Starting line.
Starting crossing line.
And does it have a marker on here?
Yes, the pilots put a waypoint on it this morning.
Did they call it waypoint 7 at 9 or something?
Let me see.
What was, which feature?
The waypoint we dropped, what's the name of it? What did we name?
The
Mound location.
Mound location. Okay.
Change
aptly
that.
named. We can change the name.
Be like,
We
deep
s-
dive here.
It was possible sample site was one of them too. I don't know. We're not the best names, I guess.
Where's your RD?
Yeah, moving over.
Thank you.
Might have heard it a couple minutes ago. We periodically hear call down from the bridge or communications with them on the vessel needing to make a move, as we are tethered to the Falcor.
Yeah, that mountain's coming
Oh,
in.
there's, yeah. That's the mound to the south of us, right?
It is to the north.
Oh, that's north of.
It's quite tall. I'm looking for
Oh, okay. Yeah. With the,
bubble plume.
oh, right. That. Yeah, I see it now.
Yeah.
I guess we don't have that much. I was looking at this, the bottom right display on my screen.
Yeah.
That's a bigger mound that we're not close enough to see.
Yeah.
But the RV is tethered to the Falcor, the vessel, and it really is the dog walking the human here, because where Sebastian wants to go, the vessel has to follow. Yep. You can see on our M3 display that orange and yellow is the high spot. Yep, this cursor marking it there. And those little black spots at the top are shadows where it was sticking up high enough to where the sonar was being blocked and couldn't get any detections behind it. So that was a pretty pointy feature. And each one of these mounds probably represents a focused spot of gas migration, to where you have enough gas to support the chemosynthetic communities, which then over time build up the carbonates and build up the mound, and then have the animal communities living there. So these seeps are really similar to like an oasis. Yeah, 3 or 4 meter high mound.
Yeah.
That's
Yeah, like as tall as Sebastian.
Yeah.
It's huge. It's definitely want to check out.
If they have time.
Yes, of course.
They have a lot of sampling to do.
Yeah, and to follow up on Jeff's point there, we sort of have these individual points of gas discharge, these little oases in the deep sea. And so one of the questions that geologists puzzle over a bit is what controls where it comes out? Why does it come out in one spot and not another? And we have some understanding of this. There are certain locations where features like faults in the subsurface or fractures or sediments allow gas to migrate through more rapidly, and they become sort of locus or the center of gas discharge. But in other situations, that's us to puzzle through what the paths that the gas is taking through the seafloor as it makes its way up to come out of the ocean, and how that can be controlled by things like the grain size of the sediment and other aspects of that seafloor material. And some of our tools here, like our sub-bottom profiler, give us some insight into that, let us see things like faults. And the coring work that we've been doing at night is going to give us some real good insight into the actual physical and mechanical properties of the sediments here. So it all contributes to tell a story about how these systems work.
Can we just keep bringing us up, or do you want him to bring us up? Should I change
You can
the
just,
range, or do you want to change?
Let's just
Leave it.
and then
Leave
change
the depth.
the color.
It's going to be less blood sage over this high-res map.
Like you don't want to have it.
It's going to get shallow, but we're going to good coverage.
For sure.
So you can use it
this morning.
and we will
I can't pull it for here.
Okay.
Because we don't.
Luckily,
Yeah, I
even
can
though
anytime.
we're changing our actual depth to the height altitude. The swath coverage gets better as we get closer because the noise gets reduced. So it's a little counterintuitive that 25 meters seems to be the sweet spot where if you get higher, your swath gets narrower, but if you get lower, it stays the same. Because the outer beams, essentially, they lose signal. So the closer, you throw them away anyway, so the closer you get, the outer beams get closer, and you get a better swath.
That's the big one from yesterday, right?
Yeah, that's the large mound that we went to at the beginning and the end of yesterday's dive. You can see there's some significant relief there.
Well, we're coming up on our last few meters of our mapping, and the mapping team is going to sign off. We've appreciated your time and attention, and we've had some really fun mapping, and hopefully it'll turn into some pretty pictures that we can put on the screen for if you keep watching. We'll get them on the navigation. And we'll hand it over, and then the next episode of this dive will be on the seafloor, back doing sampling. And then at some point in there, Adam and I need to get some rest because we have a long night of coring ahead of us.
If you keep track of what vegetable is, so you have like a if you keep track of
Thank you.
So they're not clean a little bit, but it's not a huge back there. Yeah, it's the line I'm worried about. That's because they get the careful microbes and even microbes that want.
Do we need to let Lisa know to come in, or is she ready?
Paula?
Oh, we've got a
Lisa?
couple more minutes, and then we're
Allison?
finishing the survey, and we'll go down to the seafloor, and I'd say hand over the keys, but I know who's running the vehicle.
That's excellent.
Okay.
All right.
You guys done? It sounded like.
Mm-hmm. About to click. Well, I'm gonna step away and have everyone a good morning, evening, good night, wherever you're at,
That's right.
and enjoy the rest of the dive.
A hat tip to our outstanding pilots. Yes. Our marine techs for making this magic happen this morning.
Top line. And turn it up.
Do you want a mapping entry for end of month?
Yeah. Shut it down.
Oh.
Oh, yeah.
Jeff?
Yes.
Good morning, everybody. This is Lisa Levin coming online, taking over the watch to do some sampling in biology and rocks. So I'm not sure exactly where we are, but we would like to go to the bottom so it's visible and head towards waypoint 1. Can you tell me how far we are? Okay, not so far. So we are going to go into the sampling phase of this dive, and we are headed towards an orange bacterial mat that was found towards the end of the day yesterday. So if we can stay close enough to the bottom that we can just make seafloor observations, that'd be great. Looks good. Do you think you could get patchy? Because we are going to need a lot.
I know she was. What's on and lowers off?
And also, I've worked on a lot.
Lowers.
So our sampling today will involve a number of different labs and different types of analyses. And what I'm hoping is that each group can talk a little bit about what they're trying to do. I'm here working with Eulogio Soto, who is the shipboard chief scientist, looking at the ecology of the seafloor communities, what species are there, how they're distributed, what they eat, and some taxonomy, because we are sure to find new species. Greg Rouse on this expedition is really our expert systematist. He studies the evolution and phylogenetics of these animals. And I'm going to let the two loggers talk about the kind of work that that goes on in their lab. So, Paula, you want to start?
Yeah, of course. Can you hear me? Oh, no. And yeah. Okay, awesome. Thank you. Yeah. Thank you. Okay, hi everyone. I am Paola. I am today's
Might not be
data
on.
logger. Can you hear me now? Oh. Try that. Hello? Yes. Testing? Yeah. Perfect. Okay, amazing. Hi everyone, good morning. My name is Paola and I am this watch data logger along with Erin, who is going to be presenting herself very shortly. And I am representing Dr. Turber's lab in University of California, Santa Barbara. And we are interested in this expedition on the microbial level. So we're going to be taking sediment samples and doing 16S DNA analysis just to see what microbes are driving the methane seep consumption or production. Who knows? We'll see. So we're very excited to find the seepage sites and get those sediment samples. And aside from that, we're also interested in doing fatty acid analysis, which sounds really fancy, but it really is just looking at the fat that the fauna has. And a very interesting thing about that is that on a microbial level, the fats that those microbes produce that are associated with the seep don't change that much between their trophic level all the way down to the fauna. So hopefully, since that doesn't change, it creates some sort of biomarker that tells me if they are benefiting from the seep or not. And yeah, I'll pass it down to Erin so she can explain what she's doing here.
Hello. So I am a grad student representing the Marlowe Lab at Boston University. Our lab works on geomicrobiology. And really what that means is that we're interested in what microbes are at really crazy sites and what they're doing. And so I'm particularly excited about the deep sea and chemosynthetic environments, and to figure out who's there and what they're doing, we're doing a combination of 16S sequencing, some geochemistry work to complement that, and then to kind of connect and bridge the two, we're doing some bond cat analyses so we can see which cells are active and which are not, some stable isotope probing where we can see how the carbon and nitrogen is getting used, and some incubations to see how quickly methane is being consumed at these sites.
Great, thank you. And I should mention that my specific role here, I'm sampling for stable isotope analysis, C13 and N15, and with this information at a bulk level, we often can determine which species have tissues that have incorporated carbon from methane. So I'm really interested in who can use methane, especially the non-traditional methane users. We're pretty familiar with big animals like the tube worms having symbionts. They use sulfide, but there are a few with methane oxidizing symbionts, and we're always looking for new ones. Very, very light delta C13 signatures often tell you that the carbon came from methane. So to take stable isotope analysis, we take little bits of tissue and freeze them. We acidify them, remove the inorganic carbon, and ultimately send them off, in my case, to UC Davis for analysis on a mass spec. So are we?
No, I went to waypoint 1, not waypoint 1.
We have it sorted, but we went to the M3 mapping waypoint 1, but we're headed back to the science waypoint 1.
How far is it?
100 meters.
Is that right?
200 meters. No, 100 meters. 200.
So it would be the science waypoint 1 that says orange microbial lab site. 200 meters?
200, yeah.
To the west.
Southwest, yeah.
So our ROV today is loaded up with a whole slew of different types of sampling gear. We have 16 push cores on board. For those not familiar with push cores, they are basically tubes with a handle that are inserted into the sediment, and they pull out a cylindrical plug of sediments that are more or less intact. And this is great because we have geochemists and microbiologists who need to know which organisms and chemicals are found in every sediment layer. Often they'll sample at 1 centimeter intervals or maybe 2 centimeter intervals, and it's really important to have completely intact cores that have remained vertical, haven't been disturbed or turned over. Sometimes, as happened yesterday, we release bubbles when the sampling happens at methane seeps, and the bubbling can mix up the sediments, and then the microbiologists and geochemists are not quite so happy, and so the biologists take the cores and look for animals in them. So push cores, very popular item on board. There are usually more requests for push cores than we have numbers of cores. We have to always sort those out. We also have some bio boxes with compartments in them, and they can be used to hold biology animals, like megafaunal animals, but also in this case, oh yeah, there's coral over to the left. But we'll try to keep going and just make observations. That looks like almost a paragorgia with a brittle star. We have Niskin bottles that sample water about 1.5 meters off the bottom. They're mounted near the top of the ROV, and they are used by all sorts of they're used to analyze eDNA, to give clues about whose cells are in the water column and who is at the site, which organisms. So they sequence the DNA in the water. They're also used to look at the isotope signatures of the bottom water. I do that. And also sometimes for nutrients and other measurements. There are, I mentioned the bio boxes. We use them to hold rocks, and we put 1 rock per compartment so that we can gather all the animals that have fallen off the rocks. Okay, a bacterial mat, good sign. We're getting in the zone. When we start seeing bacterial mats, it means that there's seepage from below, and almost always hydrogen sulfide, which is a byproduct of the AOM process, just like carbonate rocks would be. Here's another bacterial mat off to the left, little one. We're coming across some random tube worms, it looks like, a few tubes. And another white mat up ahead. We are looking for an orange one. The orange, Erin, do you want to tell us, does the orange indicate a different type of microbe or just a different phase of the same ones?
Yeah, I can, it tells us, gives us some hints about who's there and also what kind of geochemical processes might be happening there. So it's a good hint.
So as you can, those of you watching can see, most of the bacterial mats are white. These are filamentous sulfide oxidizing bacteria. I didn't actually look. Did you guys see whether they were Thioplochas or Beggiatoas?
I can't quite tell from this distance.
Well, you would need a microscope.
Yeah.
But did anybody look at them from yesterday?
Oh, no. Or I have not.
Okay.
That'd be a great idea though, later today.
So we're now in somewhat few plastic bags, crabs, I know, and we're at a tube bound. This is the one we were at yesterday. I'm trying to figure out, it probably is.
Yeah, it is.
Lisa, is that trash or what are these in the mats? These
Plastic
white
bags,
sheets.
trash, is my guess.
Would you mind till I get back beyond the mouth? Because I don't, I'm just trying to make sure I'm not, I can't see as much I can see right now. Yeah, go for it. Good. That's it.
So you can see that while most of the tube worms are living in large bushes or clusters that are raised up off the bottom, and that was apparently the site with the highest methane flux, there are also tube worms scattered out. I'm not sure how many of these are alive, but probably some of them.
We're just doing a quick white balance because it looks a little pretty. We're doing a quick white balance.
So these bacterial mats often have some sort of carbonate crust just below them, and so it's been really hard to sample. This is our orange mat, right? That we're trying to get to.
Is it?
Yes, I see orange mat right below us. So can we slow down?
I mean,
Is it not the waypoint? Well, let's go take a look at it. Maybe it'll be sampleable. I wasn't in the control room yesterday when they found it. Does this look like what you're after, Erin?
did you get it?
I cannot see the orange bags to which you are alluding.
Look at the front screen. It looks very orange, but maybe it's just the lights. Okay, I take it back.
Okay, yeah.
The-
no, not
So the front screen shows it as pink, and this shows it as white.
Yeah, the SIP camera doesn't have the white balance that we do.
Okay.
You can play with it however you want. Usually the closer the lights are, the more snow we get.
That was very deceiving. It looked very pink in the front. So we may come back, and if the orange mat's not large enough to accommodate the many needs, we may come back also to some of these. And there's a new find from the mapping that I'm hoping we'll have some time to go over and take a look at. A big new mound. So anyway, I was talking about the different types of samplers we have. I discussed push cores and Niskin bottles and bio boxes. We also have coral quivers, which are cylinders with a cap for holding biology.
Yeah, so that's the mouth
The orange bags.
Is this the waypoint? Yeah. Okay. So it's not very big, but we'd like to try sampling here.
For sure.
If you could possibly fire them in skin before we start collecting push cores so that we can reduce the amount of sediment. If you fire and the skin before we score,
Okay.
so that we can reduce the sediment.
Yeah. Okay, so we'll take Niskin 1 when you're ready. So the Niskin bottle provides fluids for chemical analyses that help the microbiologists interpret what they're seeing.
I can see that, I suppose.
You can see.
Yeah, I mean, you can also just write it out. Yeah. So just try.
Do we have a specific use?
So
for
when
this?
is the number one gone because it's fire? Or,
No,
Do
this,
we have a specific use?
that one's fire.
I
So
see.
the number one flipped over.
I see. Hachi? So we would call this habitat orange bacterial mat. One. I said just one. Just one, Erin, right?
Just one.
Okay, so before we sample, you guys need 3 for the Marlow lab, right?
3 cores, yes.
And Eulogia wants 5 cores around the bacterial mat, so we'll try to fit his, if your cores go in, we'll try to fit his around the edge. The Gracie Oppo group needs a bacterial mat core. And who else? I think we'll take, that's 3, 4. We'll take at least one more. Pachi, do you want a core in this bacterial mat? Do you want a core in this, in the bacteria?
Lisa, if it helps, the diet plan says 9.
I know it does.
If that,
Because Eulogia, it's because Eulogia wants his around the edge.
Okay.
So Hachi wants one, Bo needs one. We need 5 in the, we actually need 5 in the mat. And we need 5 around the edge. We'll see how we can do, if it's even coreable. So these mats are made of giant bacteria. You can often see them with your eyes. They look like spaghetti. The 2 most common types of microbes are in the groups Beggiatoa and Thioploca. The difference between them is that the Thioploca have a transparent sheath around the strands, and sometimes there's 3 or 4 or more strands within one sheath, whereas the Beggiatoa are always single strands. Usually you have to use a microscope to determine this. Another very cool thing about these microbes is they can move. They can move up and down in the sediments, and so they will move down to find hydrogen sulfide, which they oxidize, or they'll move up if the sulfide comes all the way to the surface. So when we see bacterial mats like this, we know there's sulfide lurking just below, hydrogen sulfide lurking just below the surface. That's usually makes the sediments black and smelly. This is a decent sized mat. We probably, like, I'm not sure what do you think that is lying in the middle of it? Is it fishing?
We can zoom.
Yeah, let's, before we do any coring, let's zoom, if we can, zoom in on the mat and just take a look at the surface. Sometimes we can see worms. could be a piece of wood. In which case I know we're going to want to collect it. It doesn't look like fishing debris. An old piece of tube, maybe. Yeah. Look at those crabs with bacteria growing on their backs, maybe. That's pretty interesting. You guys get a picture?
Is the fluffiness on the tube bacteria growing on the outside of the tube?
The brown? No.
No, the white. There's white fluffiness all along it.
There is? Okay.
Is it worth grabbing
The
a
crab's
crab here
eating
for fatty
some of
acid?
that, right?
Yeah. See
Let's
that?
just take a little video of this happening.
Lisa,
Yes.
is it worth grabbing one of them for fatty acid analysis, or do we want, or is it worth?
We can try to collect these crabs, but I feel like we should put the tubes in before we disturb anything. Paula likes the crabs because they're eating bacteria. Work previously by Sarah Seabrook up off the Oregon margin showed that fatty acids are a great way to demonstrate that these crabs are getting their nutrition from chemosynthetically fixed carbon. Looks to me like the shrimp might also be eating some bacteria. But I'm fascinated by the fact that there's something growing on the back of the crab. I don't know if those are bryozoans or forams or what it could be. There's also some moribund crab there that's getting walked on. Okay, I think that's good. Could we pan over and take a quick look at the surface of the bacteria? I see some little red tentacles. Those would be amphoretids. At least I saw some in the bottom of the screen. Where? Oh yeah, there we go. Right there. Yeah. That's okay, but maybe we can just pan around and take a look at the different surfaces. You can see how fuzzy it looks and how filamentous these bacteria actually are. Sometimes we see dorbilated polychaetes, but definitely there are little red tentacles sticking out like batches of 1, 2, 3, like 6. Those would be the feeding structures of the amphoretids. They also might use those to help obtain oxygen and breathe. Palps. But so there are lots of amphoretids. We can see them throughout this bacterial mat. They're below the surface and they just have their upper appendages sticking out, probably to get some oxygen. Sulfidic. They're also abundant around the edges of the bacterial mat, so Eulogio might be very happy with those cores. Amphoretids form very dense assemblages at some methane seeps to the point where we call the habitat an amphoretid bed. We've seen these in Oregon and in New Zealand seeps. They're not always associated with microbial mats. Sometimes they form a zone of their own. And it seems like they have the ability to eat sulfide oxidizing, methane oxidizing microbes because they often have very, very light isotope signatures. Okay, so everybody ready to start pushing in cores?
1 second.
So we'd like to probably try to get 5 cores in the center, in the orange part. all put in before anything comes out. And we'll probably try to get 5 more around at the very margins. So 10 in before we start pulling things in.
Right. What would get you closer than either?
The 3 of them for the Marlowe lab need to be right next to each other, apparently, I think. Right? Is that, Erin, they can't be in different parts of this orange mat? Or?
They come as close together as possible.
So, where do that clear out? Just let me know when you want the lasers on and off. First time.
Okay, actually, why don't we put the lasers on just so they can get a frame grab, and that way we'll be able to tell how large the mat is. Keep the whole thing in view if you can.
Just helping them find the lasers, because sometimes when you're zoomed out, your eyes just don't want to.
Oh, I see. We see them.
Yeah, no.
That's a great shot. So can you guys take a frame grab with the lasers, please? I'm gonna take it. And maybe a BBC Beauty highlight as well, just in case they decide mats are interesting enough to feature. The mats are one of the more photogenic features of methane seeps. They're especially for the microbiologists, but we all like them. They're very distinctive. They also form in other environments, like anoxic settings, sometimes in oxygen minimum zones, but methane seeps have the biggest, brightest, most colorful mats for sure. And that's because there's so much hydrogen sulfide there.
So you want 5 in the mat, 5 around it, all times set first. Is that accurate?
Say the last
So
one.
and you want them all set before you pull it in?
Yes.
But the 5 on the outside, can they be on the same side? Or you want
They
them
can
around
be,
the whole frame?
they can be around, well, around the edge, probably next to each other, but honestly, if,
What's easiest for us is if we do like a horseshoe shape, or just all on one side.
yeah, that's
Because if
fine.
we're reaching over
Yeah,
it, then
that-
it's risky.
That's fine. And it's okay for the edge ones to have some bacteria in them, I think, yeah. Now, if we run into hard stuff underneath, then we have to reconsider what we do. So we'll just see how the first one goes.
So what if we run into first? This is our mission control.
See all these screens. So we have our science lead. We have one of our pilots. We have our co-pilot here. So we got Alex and Sonny. We got our marine technician. So up front we have 4 people doing very important jobs. So we can look at our Sonny. So we talk about the push course that we're doing. So right now, he's using a manipulator. It's a robotic arm. It's not one of the cylinders to push core. It's going to drive it into the seafloor to get a sample. And so the image surface is rolled up. You can see Sonny operating.
So we never know whether the core will penetrate fully until we give it a try. It's one of the hazards of sampling at methane seeps is that the microbes create a lot of carbonate in the sediments, and sometimes there's a carbonate crust or carbonate pebbles that block the core.
So the control center is a very small version of that large robotic arm. So when you move something on that small version of the arm,
Somebody's asking if there's methane vents here on the chat. And the answer is yes, there's methane seepage. We call these seeps because it often comes out very, very slowly without gushing out of a vent like chimney. Occasionally we'll see it gush, but more commonly it bubbles slowly. Sometimes there's no bubbles at all, like right now. Yes, I would say try to get it over the colored part rather than the brown part. Yeah.
Bueno, por acá, gracias Muñoz, de la Universidad Católica, solo para describir lo que estamos haciendo aquí, tomar algunas muestras de sedimentos superficiales en la zona donde están los restos de bacterias, que son de color naranja, probablemente que bloque. El sedimento se ve muy borroso y esperamos poder tomar unos buenos centímetros de sedimento. Como indicaba eso recién, a veces no es fácil tomar las muestras. Aparentemente nuestro estado penetrando bien, se sedimento bastante grande, pero a veces quedan estancados, digamos, porque no las rocas carbonáticas que se forman en estos sistemas generan dureza en los sedimentos y no dejan tomar bien profundos los testigos.
You see bubbles coming out, so there's definitely methane under there. Can you tell how far down that is? Aaron, can you, or you? Right, halfway. Halfway?
Okay.
Is that gonna be okay?
Yeah, that'll be alright. I'm more worried about getting at orange than that. No orange there.
There's definitely methane. We see bubbles.
Is there a response in SAGE?
No. Well, we won't see it for a bit if there is one. But when we had bubbles yesterday on coring, we didn't have any meaningful increase in methane.
Okay.
And I wouldn't expect it. You need something more substantial than that to actually get the water column elevated.
Okay.
Yeah.
Scott, would it be already set up to possibly stick the temperature, I mean, the methane one into the sediment?
We cannot go on to the sediment.
Okay.
That's like not even like.
No worries,
That's not
I've never checked
yeah.
that, but I'll let you know. Okay.
We aren't connected to one right now. I just texted the chest.
Oh, okay.
The chest acid.
Okay.
The wand, yeah, the wand.
When did you text me? No, when? Or am I did?
Yeah.
Because you texted me something yesterday, it's kind of confusing.
Oh no, I wasn't texting about sediment. I was asking if he wanted the wand or not. He was saying pass it for now. You know, kind of if you switch over if needed. I'm not sure now that he realizes that the wand is water safe.
Yeah, I don't think he does, so I'll let
I'll
him know.
I'll
Okay,
shoot
thank
him a
you.
text.
When that first push core went in, you saw some black silty material come out the bottom and waft away, and that's that very dark color tells us there's a lot of hydrogen sulfide down there. Microbiologists like to have cores right next to each other. They do different things with each core and need to have them be fairly intercomparable. There's a lot of spatial heterogeneity at seeps, as you can see from the patchy bacterial mat. So one core a meter away from another could be sampling a very, very different environment.
That's a good location, yeah.
The surface of the mud looks almost like jelly. It's very, very soft.
Which part, or say it'll come back up, just maybe rotate it.
Just close bit the
Yeah, but you don't wanna.
plunger for the other one. Top of
What's happening here?
the
So don't, so next time just.
Because it's going to disturb the whole rest of the mat if we take it out right now. Are you going to be able to use that, Erin, or not? No, we if we shake it out, we've sort of destroyed the mat.
Yeah. If it would be possible to take the Marlow cores on this side now, that would be ideal, because we're looking for depth gradients, and so
So do a
if
flat
the
on the other side?
if the reduced sediments are on the surface of these, we'll get some excursions.
Okay, so let's just. Probably, do you want to put that core back then?
Well, we need to shake it out if it's not a good core, but it needs to be far away from here
Yeah.
that happens, so that it doesn't destroy more mat.
Yeah,
We need to try to get 5.
Then it's just leave that one.
Leave the one that's still there, one.
We'll leave that one too, don't use it right now. We'll go 5 clean ones, and then we'll shake that one out after we're done, so that way we don't risk disturbance. Just like put that in the center, just to store it.
Well, it's almost perfectly clean. There's only one thing sitting in there. Honestly, you can shake it out probably. But is that a problem, Erin?
If you can shake it out, it's fine.
I just think that they'll have to go in fast to avoid releasing a lot of stuff.
Once you come back up there, just put it. So if you can reach that, great, otherwise it's not dry.
I'll take it, go to the left.
Well, they need 3 together and in a very clean part of the mat, and we've already covered half the mat with mud, so.
You said 5 in the right part, correct?
No.
Personally, I'll need 3 on the right part.
Okay.
Together.
And 2 other people need bacterial mat cores.
Yes.
Hachi and
Yes. Does that include this
David,
one?
and that can be one of them, but
So we'll need
we
3 on
can't
the side
sample
and
the
then
black, the place where we've had sediment go over the top, yeah. So we'll see how many we can fit in this right side. And if we have to, I'll get, Hachi, you want this map, this site, right? Can you use that first core?
If you need, just tell me before.
So something that they noted yesterday was there's often a carbonate layer.
Yeah.
like Pachi's telling you she can't use that first core because it wiggled.
Yeah.
So if there's not, if it's too hard, we can't
Yep.
use these sites.
Yeah,
So
just-
they need to go straight down without wiggling as much if possible, and we'll just see. So Erin, if it's hard and we can't get good cores, you want a different mat?
If you can get at least like a half core, I would still take it. If it's too hard to go in very far at all,
Yeah,
then
so half a core is okay.
Given where that plume of sediment ended up, it might make sense to go to bottom right most, because stuff's gonna come out when you core it, it'll float that way.
So start on the bottom right part of the patch and move
How
up.
about we do this, how about I'll put the lasers exactly where you want it, the left laser, and then you'll go right on that laser.
Okay.
So tell me where you want it, guide me to the left laser.
yeah, I think that's good. I think that's a good idea, Scott.
Might be before you start, you might just kind of travel over your screen.
Okay.
There it goes. So that was the current presence.
When sampling like this, we always want to have a little bit of current so that the sediment clouds move away. But too much current makes it very difficult to keep things stable.
Yep.
It's really interesting to see animals eating the bacteria. People in the chat are calling it sherbet, but I think it's anything but sweet.
I know you're using it this week.
But when we see the shrimp and the crabs out here eating the bacteria, it helps us understand that methane seeps are a really important part of the food chain for the entire ecosystem, not just the seep ecosystem. The dark spaces inside the mat are really interesting because very often they reflect grazing that some organism, a snail or amphiridids or some other animal, maybe the crabs and shrimp, have actually eaten the bacteria and left these dark patches.
Is that laser okay?
Yep.
Almost.
Ooh, that's nice. It's good. Out come the bubbles.
But without it, just realize it's something like that. That's your whole way to do it, so it doesn't stand out.
You're gonna want to try to get at least 4, 2 more in there.
Okay, it was just 3.
Well, it's 3 for her, but we have other person who needs a core next to theirs, so that's
So-
Pachi. Yeah.
So next time, just tell us how many total in the area.
Well, 5 in the bacterial mat.
Right, but like in this side.
Okay.
So that way he can plan out where they're gonna go ahead of time.
So Erin, that's also really important when you're taking your 3 cores, there will have to be others with them.
Yeah.
And they were under the impression they were only putting 3 in right there.
Just look at them
So-
on your right, right?
Yeah, you should, you might be good. You can see it on the page too.
Down.
Can it be, Pachi, can yours go in a different part of this map?
Could be
You just probably get the right just-
side, the left.
We'll still try to get the
You take, yeah, go ahead and.
Look at what's it.
Yeah.
So,
Between 2 and 3. There are no space.
you okay with that, Erin?
See, like,
If you go up a tiny bit just to avoid getting some of the reduced sediments. Yeah, that'd be good.
might be able to reach your last one.
So Paula, the crabs have kind of walked away.
We'll find them.
Eulogia sent a message to make sure we got crabs.
Someone
But-
was on the shelf. I don't know who it is.
You might have to meet as well, those steps.
It's, hang on one second. Thought you said, can they do the lower part of the mat? Come here, why don't you show me? He has to grab it a different way.
The other arm.
I'm sorry?
We're using the other arm.
Oh, they're gonna change.
Alright.
The handle looks like it. And will we be able to use that arm to put in the cores that go around also? Of the other five?
Yeah, we can use either arm to get the rest of it. That's it.
Okay. So Erin, we're gonna put the Eulogio's course in around the map before we take anything else
Yep.
out. Yeah. But this one's for Pachi, yeah. Somebody's asking what's the difference between the arms, and it's pretty much what they can reach. So if we ask for a core in a very specific place, it's often reachable only by one of the two arms. So they switched out for this next core. Are you okay with this? Oh, I do see a crab in there. Great. Okay. It's a nice deep one. Very nice. Okay. If we can put 5 around, somewhere around the edges.
If they were all just like here.
Yes, I think that's fine. You love you.
As far as you can reach, I suppose, like practically, up here and then back towards this way, we'll probably want to put this one first. Get that towards the end. And if you need me to do this up.
Does anybody know whether the Oppo lab core, Gracie's core has to be perfectly intact? Can they need a bacterial mat core, can they use number 1, do you think?
We can ask them.
We'll see what comes back. Let's see which we collect, okay?
So this next one would be great.
No, this next 5 are for you, Lobio. Yeah. I was asking about whether number 1, which could be for them. I think these don't have to be in a very precise place. They can just be close to the mat and around it, wherever so you can reach. Wherever you can reach is.
It might be better if we refer that we
Well, don't move out away from the mat, just around, if you can get around the mat. Yeah. Yeah. Great. That's good. Stop. Not too full. Do you want to be far off the site, or you just want something that's not that?
Further off from the site.
Okay. Yeah, that's great. Hmm. It's probably deep enough. Yeah, it's fine. Preferably not over a shrimp. I think I'll get out of the way. So there's a lot of different kinds of coring going on this expedition. The push cores are the smallest. They're 7 centimeters in diameter. But they are by far the most precise in terms of what's targeted. So when you have an ROV with push cores, you can take samples as we are now in different parts of a bacterial mat, right next to a bacterial mat, away in background, and you can see exactly what you're getting. It's much, much harder with the other forms of cores we have on board, the multi-core and the gravity core. The multi-core, I believe, has a camera on it, but it's still fairly difficult to position it the way we want. So push cores are a hot commodity, lots of demand for them. There is a discussion in the chat about experiments. They happen all the time with submersibles and ROVs. You can move things around on the bottom, add things to the bottom, put out labeled material that you collect later and see what happens to it. It could be labeled phytodetritus or something else. So lots of experimentation goes on in the deep sea these days. Very common for those who study wood is to put out intact wood blocks untreated so that they're natural and come back and collect them later and study the wood boring and wood decaying fauna. Even whales get towed and put on the bottom for people to study the ecosystems that develop around whales. I would say they're already dead, and they strand, and then some scientists will deploy them onto the deep sea floor. Maybe away from that tube, somewhere not near it, just in case it gets in the way underground. If you can move a little closer to the mat, yeah, that's great. One more. Thank you.
Where do you want this one?
Next to 6 or 4 of, yeah.
That's it.
Is there any place you can, I mean, it can go between 6 and 7 if you need to, if that's reachable.
Yeah.
Or between 7 and 8, but.
Yeah, it's okay, right?
Yep. Where do you want your second floor? You want it somewhere else? This floor too. Not here. Okay, great. Let's just get a couple of good shots of the juxtaposition. I mean, one of the things that I think we should take the cores out first, but we're, just so you know, we're going to be probably want to suction this mat, get a suction sample, and we're really after any of the crabs that are here. Most of them ran away, but if you see any crabs, let us know. Yeah.
There's one still behind you, coral there.
I think it's dead. Do you want dead crabs?
No.
It hasn't moved.
It hasn't moved in the whole time we've been here. I saw that one, yeah. It's unclear if it's alive or dead, but probably dead. Yeah.
It's got quite a lot of bacteria on it.
It's not the most hospitable setting. Okay, I guess you got, there's one, yep. Would that one be okay?
That would be fine.
Yeah. After the cores go in the, I just don't want to risk knocking cores over or anything like that. So.
No, some of them didn't go all the way, like one, two, didn't go all the way.
Hey, Joe.
Ready to start removing cores?
Yes.
Okay.
Yeah, and, you know, they can go in any space that's easy. You don't have to put them in order
Right.
or anything like that.
Okay.
So whatever you can reach best and get them in the fastest so the contents don't fall.
Gotcha.
Yeah.
It's the race, isn't it? Yeah, may as well get the close ones.
Okay.
Get them to roll furthest away.
Well, here's the thing, what you don't want to do is you don't want to drag cores across open. So if you drop it into one, it's gonna mix with the next one.
Right.
So,
Right.
you wanna fill up down
Right.
here and then fill up down here and then, so you wanna go farthest to closest.
You're right.
This way you're not dragging it over the next one. So that's why she said it doesn't matter where we put it. So whichever one you wanna grab first, put it in the very first one.
Got it.
Yeah. Just making sure we got the theory down.
Yeah. And it's one of the reasons, like, why I haven't filled in anything yet. I'm gonna wait and see what, we're gonna wait and see where they put the cores. And
Which one are you going for? Go for 10. Okay, that was the last one you just did.
it doesn't, time-wise, it doesn't matter, so whatever's easiest. ones in the mat are the shortest and probably the most likely to fall out too. So I don't know if that matters where you put them. But.
We can just put them in the easiest place then.
Yeah.
Up front. Go for number 2 then.
Okay.
Let's see, I'm curious now.
So, tilt a little bit.
Excellent. Thank you.
First course of the hitch, right?
That's right. Which one you go for next?
I'll pick up 4. Try to get 4.
Okay.
That'll clear the way for 3 work.
How are you doing today, Lisa?
I'm good.
Good.
How are you?
Good. How did some of the stuff you got yesterday?
They were
turn out?
great.
Yeah.
We're still working on them.
Awesome. Okay.
They're excellent samples yesterday.
Good.
Very exciting to see so many things at this Seep.
Yeah. How did those ones come up that had the bubbles in them? That was kind of interesting.
I didn't see the
Didn't see
push
any
cores. I didn't process push cores last night. I was dealing with all those fuzzy rocks,
Okay.
which were
Did you get some good
great.
ones?
Yeah, we
All
did.
right, good.
Yep. You know, prior to our dives, there was like about 3 or 4 fauna types of species listed from this site, and now there's probably 100.
Wow,
I don't know.
okay.
There's a lot of things here.
Oh, no, it doesn't look like it's gonna stay.
Looks unhappy.
That's all I can do in the bush. That's definitely falling out. There you go. Just push it down. Yeah, I think so.
So Aaron, that core is not gonna be too good. You wanna try to use number one?
I'm texting about that right now.
What?
I'm texting Jeff about it.
Okay. Or we might be able to punch another one. But somebody else is gonna have to give up something. Can you live with 2 background cores or? No.
Or he does not need any more cores.
Okay.
So.
Yeah, we might be okay with the numbers. If Greg and I don't take it. It's like.
I think she's checking with Jeff to see if it's good. If we could try sticking another one behind 3, that would definitely be great. But I'm just texting Jeff about whether it needs to be.
Well, go ahead and let's collect 3 and see how it is, because we could easily have some more with problems. Let's just see what happens. 3 is next to 4, it's the same sediment, so. And there's bubbles coming up inside the core. Maybe they're from the outside, I can't tell. The core looks pretty intact.
There's a bit of gas within the
This one comes up with a stick. Wherever it's easiest, now I can put in the red one wherever you want. That's the one we have. Just watch when you come across the top of that. Yeah, there's bubbles in there. These are top of the core.
Yeah, interesting.
We had something like that yesterday.
Maybe a gas will form gas hydrate or something. See the bubble in the very top of your core? Yeah. You could sample it.
Hopefully.
Maybe it'll make its way out the
Doesn't
I guess
expand
the
all the way
solution
up.
was right
Top
now,
on the
but
top.
not.
Okay, so do you want us to try punching another one?
Have not heard that. I'm texting him right now. If you want to collect botches in the meantime, that's far enough away that I don't think it would cause any.
No, they might have to move. Or switch arms, I guess.
What's that?
No, any of these shouldn't disturb the.
What are you asking?
Well, we're thinking we might want to take one more core in the back part of that mat, right behind.
Okay.
But
After
while
we
she's
pick these
waiting,
up?
she's contacting Jeff, so she's just requested that you collect cores in somewhere that won't disturb that spot.
Okay, so any of these 6, 7, and 8 won't disturb
It
that
should
spot.
be fine if you come over this
Got it,
direction.
those.
Probably 9 is the most likely to create. number 5, which isn't showing, is this one. That's probably the one where for geochemistry, it's also the most important not to have it disturbed. So if you want to put it like right at one of the front ones.
You could go for 6 if you want.
Or 7, or, you know. Today was Jeff's baby day.
It's
You might-
tomorrow now. Yeah.
It's tomorrow now?
Yeah, it's scheduled for tomorrow.
Oh.
He said trying a different core up here would be better than using one.
Do you think Gracie can use number 1?
I can text her.
Yeah. Or she can wait till tomorrow even.
Jeff's also suggesting that the core that failed, number 4,
Yeah.
we could shake it out somewhere else and reuse it so that no one has to move out the core spot, if that's helpful.
There's stuff in the bottom of that quiver now.
Okay.
Wouldn't that bother somebody?
Yeah.
So we do want to try punching another one? Do you think it would be a good time
We'll
to
get
put
7
one
out of
in?
the way.
Or just, okay.
We'll grab the core next to it, put it out there.
Right, so the next empty one is 11.
Starting to slide, just nice and easy, nice and easy.
If Gracie can wait, or if we can't confirm that she could use one, like number 1, we could also shake that one out, reuse that one for this back core. If she can wait on the core, or if we don't know, if we're not confident she can use
just said it's served more anyways.
You think it's better to do that? Shake out number one?
I think it might be.
Are we putting another core in?
Well, there's a discussion, because we're so short on cores, that we might want to shake out number one, which got somewhat disturbed and wiggled on the way down, and
So you
use
wanna
it, use it as that extra
Why
core.
don't you just grab these two now?
Yeah, why don't we go ahead and do that? And actually, and also the geochemistry one's pretty important, that one down in the corner, so. I mean, we can also use that disturbed core, so maybe we should keep it for animals from the bacterial mat. But actually, if we're gonna section the mat, it doesn't matter. We don't need the core. I would like to just get some of the amphorithids and anything else that's in there from near the surface, so. So I think, Erin, we should take a clean core and
Okay.
put it in,
Great.
yeah. Great.
You can grab 9 with that on if you want.
Okay, so we're not sticking out.
I think they're just worried about it disturbing that clean part of the mat.
Oh,
over there.
are we taking another one back?
Yeah, we're gonna take another one back there, and
Yeah, switch
actually,
arms.
we'll have you use 11 or something, a new core, not shaking out an old one.
You wanna put it in now?
Yeah.
Or shall we grab 11? So you can stack it back there.
So the bacterial mats that we're sampling, as you can see, they're really patchy on the seafloor, and they really, oh, look at there's somebody came for a visit.
That was cool.
He was trying to eat your manipulator. Oh my goodness. That was a Humboldt squid that just made an entrance briefly, thinking the bright red soft claw on the manipulator was some delicious food item. Maybe a shrimp.
Something clickbaity for specific.
11 is empty, right, guys?
Yes.
Yeah. What just happened there? Did something fall in? Or somebody fell in? I mean, hydrogen sulfide's pretty nasty stuff. It could stun animals.
Oh yeah. Is this a gradable spot right here?
Yep.
It looks great. Uh-oh.
Okay.
Back in the quiver? Yeah, sure.
Are you happy with, you need a far away picture? Are you happy with that?
No for that. Okay.
You're good with this core, Erin. It can go right in the quiver, right?
Yep.
Yeah.
And contact with, not coral, but
So this was 11, and it's going right behind here. One of the things about watching the coring operation is that it's extremely difficult. Typically, the pilots make it look easy, but it's not. And when they have finicky scientists who really want undisturbed samples that haven't been tilted or bent or anything, makes it even harder. So you're watching a lot of skill here. It's a great view of the filamentous nature of this surface bacteria. Very nice, thank you.
You wanna get 9 while you already got that arm out?
Yeah.
You happy for us to take 9?
Yep.
Okay.
Just leave a good spot for 5 because that's the one we.
Well, why don't we-
5
why
is
don't
the
we
lower
get 5
left
Yeah, let's get
one.
5 in. Whatever Sandy is.
Can it go in one of the front left ones or?
It's almost direct line up there. Yep, and then we'll do a graph
So the strategy here is so complex because they actually have to figure out which open quivers are closest to which cores that are in the sediment to reduce the likelihood that the contents will fall out on the way in to the quiver. So there was some strategizing there. Well, here you don't need any Niskin, right, for your samples.
Just on top of it. You see it, the sit, there is about touch the top of the bowl. Higher on the other side.
Pachi, you want to talk a little bit about what's going to happen to this core that they're picking up at? We happen to have the user of this core number 5 with us. She's going to say what's going to happen to it.
Está relativamente sobre el mar también. Entonces ahora la maniobra es tratar de sacarlo lo menos perturbado posible. Confiemos en que sí. Dios quiera que sí. Se le ve los mats en la parte superior. Tiene algunas zonas que se ven claramente anóxicas, producto de las condiciones reductoras. Y perfecto. Bien, parece que ya podemos respirar. La idea es, en estos testigos, sacar el agua intersticial, la que está entre los granitos de arena, para medir algunos elementos como el sulfuro. En mi caso también vamos a analizar metales. Y de la parte sólida, isótopos estables, también vamos a analizar algunos elementos químicos.
We're going to want to suction the surface of that also, so you could even do that before 9.
Sure.
Bueno, realmente es importante entender la geoquímica de los elementos en esta zona, en el caso de los metales, porque muchos de ellos son utilizados como indicadores para estudiar las condiciones oceanográficas.
I don't know, it might be easier to get all the cores first and just have it done, but so we don't have to worry about knocking anything over. No, but we just came right from the end of the survey to this mat without going by much, so we did not see it. But we haven't been over to the main mound area yet. Or maybe we crossed over it. I think we did cross by it. I did not see the quivers on the ground, but do we have a waypoint for the
I have
paddle
a marker
quivers?
where I think you can find three.
Yeah. Okay.
Wherever you want to put it in China.
Bueno, probablemente en ese punto específico hay carbonatos que impidieron que el testigo pudiera penetrar un poco más.
Yeah, put it in that one. It's all right. Just a little chunk off. You're good.
Might go to Gracie. Might go to Gracie. She can use it, or we'll take it for fauna.
I think you can use that number 9 with that on, and you can see.
It just,
Oh, yeah, you're-
the surface doesn't look very disturbed.
Nope, you're in that core. Yeah, I think rather than the other one. I don't know. Reach across the core, just stick it in.
We could move, can we move the vehicle?
Yeah, we can move. I'll move to get getting closer. Alright.
But, yeah, just as long as we can keep some of that mat suctionable. It's interesting to see who does and doesn't like to hang out around the bacterial mats. We know scientists like to, but the fish seem to avoid it. I haven't really seen, has anybody seen any fish swim by or swim in? I think they don't like the hydrogen sulfide there.
There's been a skate behind us for the past hour or two, just right behind the vehicle.
Okay, so the skates are pretty tolerant then. Yeah,
Maybe it's far enough away that they're not like it.
yeah.
That's gonna be.
But the crabs and shrimp seem pretty happy to hang out around the mats. As I said, these are areas, usually we identify the mats as areas of relatively high seepage. However, our methane master back there tells us that we don't really see, we haven't seen any elevated signal, right?
Only when one associated with the bubbles that came out of the core, but
We did see the bubbles show up.
Yeah.
Yeah.
One of the two sets of bubbles, not both.
Okay, yeah.
But Lisa, I'd say the baseline is a little bit higher, but there's not hugely elevated methane here.
But there is probably quite a bit of methane under the sediment, given that when we poke it, bubbles come out. It's pretty exciting to have a methane sensor on the ROV. That's not been the norm, even for our other methane seep projects. So, and we do know there's a huge methane signal over the big tube worm mound. So we're waiting for the sediment to clear. We have one core left, right, to pick up, and we'll try to do a section.
And after that, we'll try to get the crab.
And then we're gonna go for crab. Yep. Thank you for the reminder. You want to talk about what you might do with that crab?
Yeah. Of course. So there was, as Lisa mentioned earlier, there was an article by Dr. Seabrook in around 2018, and it showed surprisingly that through biomarkers, especially fatty acids, we are able to compare and correlate the energy that comes from the seepage and the crabs that are actually benefiting from it. So it was a direct link, and it was highly significant. So hopefully what we're trying to do now is replicate that at the Seep. So for that, I usually only need tissue samples, so even one leg of the crab is more than enough. But hopefully we get the whole thing. But yeah, that's what I will be doing, and if I'm correct, then the sample would stay in Chile, in the museum for collection.
We've got Peter on now. Peter, do you want to talk about what's going to happen to those 3 microbial mat cores that were just taken?
Sure. We're going to be doing some incubations with artificial amino acids, and these amino acids will allow us to later use fluorescence microscopy to determine the activity of the microbes, which is really important because microbes can enter dormant states, they can enter active states, and being able to determine if they are active allows us to help understand which microbes we should focus on for things like bioprospecting. Oftentimes these microbes are transforming gases that are not very helpful for us, for like methane and hydrogen sulfide. And if we can learn more about how they transform those, maybe we can use that to our own benefit. We'll also be looking at the microbial diversity and storing some live mud for future analyses as well. Thanks, Lisa.
Sure. Great. If our group ends up, well, or number 1, we're not sure who's going to end up with it. There's a professor who's not on board, Victor Gallardo, who studies really big bacteria, macrobacteria that you can see, and he might end up with the bacteria in that core. We're kind of interested in the animals that hang out and eat those bacteria. So again, I'd be doing stable isotopes on the amphiridids or whoever we get out of the bacterial mat. Some bacterial mats are full of dorvalaid polychaetes, but for some reason we've seen very few on this expedition. This last core here is Eulogio Sotos. He's not in the room, but he'll be doing the ecology of the macrofauna and myofauna in the sediments. Those are all the small crustaceans, polychaetes, annelids, cnidarians that live in the mud. He'll be doing quantitative analyses to characterize those. I'd ask the pilots to talk about themselves, but they're really pretty busy and have to concentrate on what's happening here.
Hola.
Ajá.
Hola.
Hola.
¿Tú sabes cuáles son los músculos para mí?
Sí.
¿Qué números son?
6, 7, 8, 9 y 10.
Esos son para mí.
Son todos tuyos. Y este es el último, han corrido su turno.
6, 7, 8, 9.
So this is Eulogio's 5th sample. The strategy today is to get all the cores taken early so there's less angst and anxiety among the scientists for the rest of the dive, where they can enjoy Greg's description of what we're seeing, and he can do more exploring. But we still have a handful of background cores, one more for Pachi and 3 background cores for Jeff to take. So we have no margin of error given what's left empty. Do you think we can section the crab, Paula?
Sorry?
You think the crab can come up in suction sample?
It look small enough.
Yeah.
That'll work.
Great. Thank you. Yeah. Can we see the map? Barely. Did the current change direction, or we just changed direction?
Get set up to pick this core up.
Okay. So is it coming at us now?
It's kind of like just, as you can see, it just blew it right in front of you.
Yeah. So.
Try and move again, but it's gonna
Well,
sit more.
maybe we can, is this enough visibility to try suctioning the
Yeah,
mat?
you can see the map.
Yeah. So we'll try suctioning the mat, and then we want to get a crab. But the question is, will we still be able to see to get the crab? Okay. I don't know. So we want to use one of the finer, probably the number 1 or 2 for section.
Looks like we're lined up at number 1.
That's great. I assume that the microns haven't changed.
See the nice soft handle?
Yeah, I like that.
So we'll just go for the upper centimeter or 2 of the sediment. Don't stick it down deep into the holes, for example, please. Yeah, most of the animals that we would be after live in the upper part. So.
Yeah. Something quite in the animals.
Which suction sampler are we using?
Number 1.
Thank you.
We're hoping is that we get some of the worms that are living in the sediments here, along with the bacteria.
See right there, where it's orange. Get a good bit of the top, like right across here, and then out there.
It's okay to go down into the sediment a little bit more. Yeah. Ooh, is that a fish? Oh. That went by? Something. So this suction has a 540 micron mesh on the canister 1, so we're hoping we'll be able to retain some of the small things that might be in the sediment.
Nice chunks there.
Nice, those clam shells under the mat.
Yeah, I can't tell if it's fragments of clam or if there's whole clam. Oh, well, those look like whole clams, don't they? If you can get the clam to go in. Well, okay, that's a broken shell. Yeah. One of the things we found is that clam beds, which are characteristic habitat of seep, sometimes get covered over by bacterial mats. If the seepage increases, the clams could die.
Ooh, you almost got that right.
There's a tube. Try to avoid getting the tube in there. I think it'll get stuck.
We could take some clams samples.
There's definitely shells underneath this mat. Can you tell if the canister is full?
I can't because it's got a bunch of dirt in it right now. So you see how dark it is? We'll have to actually move away for a bit and let it clear.
But I think that's probably good enough.
Okay.
Oh, polychaete just fell out. Yeah, that is a fish. I wonder can you get that fish? That would be quite, it looks like a snail fish to me, honestly. So do we need to run it a while, given how long the hose is, to make sure everything gets up?
Yeah.
That was a strange
I saw the
fish.
fish hit. So it's good that we got pretty much everything in there at this point.
Okay.
Go ahead and
Great.
shut down and index. Are you gonna want the crab at the same one, or
jar?
Probably different. If we can find the crab. So I guess there was a crab hanging out by the tubes.
Yeah, I can see one just scuttling away from me.
I mean, if you felt you could pick them up and put them in a coral quiver, that would be fine too. But yeah, whatever's the easiest way to get it.
You see it, you can see it scurrying away over there. I'll give chase. Alright.
Are we following a specific crab here?
Yeah. It's on jar 2 though. Which jar do we want the crab in?
Probably 8, 7, 5, 6, 7, or 8.
This is an acceptable crab right here.
Is that the right kind of crab? Can you tell, Paula?
It looks like it's.
Okay, I'm trying to look at what it is. Yeah.
Yeah.
That's one of them.
It has
Little
the-
spider crab.
Which suction sampler are we at? 8?
It was 8, right? Number 8.
Yeah.
Thank you.
If we see another, anybody see another one out there? It'd be good to try to have 2 in case one disappears.
I will appreciate that.
You want the snail? Okay, we have a request for that white whelk.
Same
Right
jar?
there. Yeah.
Okay. Suction's still on, buddy.
There's another one of those fish. Don't those look like snail fish? Like parrot? Hmm. Could we? Well, I was gonna say, it'd be nice to zoom in on the fish at some point, but we don't have to do it right now.
There's a fish.
While we're
and
sectioning.
bacteria.
There might be some crabs hanging out under the tubes, the cluster of tubes that was over on the other side. Oh, there we go. This really looks like a liparrid, which our common name is snailfish. They are one of the fishes that are special, well, they live from the intertidal down to the deep sea, but they are the deepest living types of fishes. They're found in trenches down to about 8,200 or 8,300 meters. And they lay their, some snailfish lay their eggs in xenophyophores, which are giant agglutinated protozoans.
All right, what's next?
Did we stow that?
No, we haven't.
Okay, then, well, if we have enough visibility, let's look for another crab right around here, because I think it would be safer to have 2.
I agree.
And we're specifically looking for if the crab has signs that they've been eating bacteria. So the tube worm cluster, I think, had a crab hanging out under it. But we'll take any one right around here. Oh, there's one. Right here. Okay. There were a whole bunch of them on the orange bacterial mat when we arrived at this spot, and we scared them all off. So we're just re-hunting them. There's another, right there. There. What is
Oh,
it?
there are three. This is the species we want.
Yep, they're
Same
both.
jar?
Yep.
Alright. Sure, the bigger one. Do you know bigger one fit? Bigger one might fit. Let's see how big you are.
Like it's stuck in there.
Is it the small one will fit?
The small one will fit better, but
Not the big guy?
either, whatever you feel will work.
Who's going for that shrimp? You gonna get a surprise. Oh,
They're fast. Bye. Great. Amazing. So which canister did you end up?
8.
8. Okay, so it's
There,
got
one.
2
See
crabs
that one, he's coming our way though.
Yeah.
and 1 whelk.
You want a bonus crab or rich in crabs?
Did
right?
you get?
If it's close by.
There's 2
then-
of them there. They're gonna face off. See them?
Yeah.
Case.
That big one's in the jar.
Sweet.
See if we can reach them. Surprise them.
Yeah.
With their buddy here. Oh, you got shrimp too? Oh, man.
Amazing.
Now we got
Thank
some.
you.
So we got 4 crabs and a shrimp, right? We could have lunch.
Yeah, I can have lunch now.
Anything else? Any other shrimps? Crabs? Fishes?
Okay, I think we're good here. Can I ask you how far away the new mound site is that they laid a marker for and just called it New Mound? Is that right?
It's 100 meters, yeah, it's to the northwest. There you go, mound location.
Okay. So what I think we'd like to do is head over there, and if, Peter, if you see a background site that's background enough for coring, we can try that. Hachi, if you see a site where you want your second core, you can tell us, okay? You want it in a seep or in the background?
In the background.
You want a background core. Okay, so we would be doing 4 cores, probably our last 4 cores in the background. But I'm eager to see the new feature discovered during the mapping exercise at the first part of the dive. And we did leave an orange bacterial mat for some future scientist to come back here and pillage. Just for the record, 1 undisturbed orange bacterial mat remains. Look at all those shrimp. That's it.
Internet official, it's on the web.
Yes, well.
All right, where are we headed?
So, do you see a waypoint called New Mound or something along those lines?
Oh, we're going all the way up that way.
So it's 100 meters or more?
Already.
Or is it 1,000 meters?
No, it's okay. Okay.
We could do that. 1/3 of a football field, right?
Just have to move the vessel with your buddy.
And if we do see anything that looks like a clam bed, we should mark it. Like, we've only seen dead clam shells so far.
Yeah, go ahead. Hey Tony, that's a new waypoint for you. Copy that. Thank you.
All the white on the hill here, does that all look like dead shells to you? Because I know we're really interested in getting live clams. But we've got some tube worms.
That looks like a pile of two worms.
Maybe this is the other side of our big mound or something. Greg Rouse is interested in the fact that the tube worms have different morphologies out here, that there might be more than one species. What is a big fish? What is that? Is that a dogfish? Dog shark?
Looks like
Lisa, I think that's the mound that was yesterday's waypoint 2.
Okay. So in theory, we've surveyed that. So if we, on our way, we find something where there's no seepage and no clamshells and no tubes and no nothing, that's our background site, is my understanding. And we might want to stop and take 4 cores. There's lots of shrimp. They seem to be part of the background scenery. Lots of burrows. We don't know who's making these burrows, but typically they're made by either burrowing Callian acid style shrimp or sometimes echierans, which are another type of worm. I think are classified as polychaetes now, I'm not sure. Definitely a hummocky bottom, meaning it's well bioturbated, probably, with lots of, you can see little openings at the tops of the make these depressions to trap food, and then they'll feed. There's our skate ground. You see an octopus?
No, it was the shadow of the fish.
Grenadier. My daughter loves skates and rays. She has tattoos. Every time she sees a new one, she gets a tattoo.
Is that right?
I should come home with a
There you go.
tattoo
That's
of
right,
this skate.
yeah. That would surprise me.
It would.
How many skates has she seen so far?
Has she?
Yeah.
Oh, I don't know. She's an avid scuba diver and goes all over the place and sends beautiful pictures of skates and rays. I assume she's not listening. She's at work. Most likely. So word has it this new site is a big mound. Was anybody in here when they mapped it?
Yeah.
And what did you learn about it?
From the multibeam, it showed about a 3-meter, very pointy.
High reflectivity.
Object. Yeah, high reflectivity. Jeff thought it might be a bunch
Once
of
we do actually look
two worms.
Okay.
No.
Let's hope it's not like a container that fell off a ship or
Yes.
something
Right.
like that.
If it is, hopefully it has good stuff and we can recover it.
Maybe. Okay, well, we're sort of back in the seepy realm, so things are getting, there's more tube worms here. This is a really fun part of our job, exploring new things that nobody's seen before. Something up ahead, right?
Huge. It's,
Maybe we should slow down and not run into it. A lot of waste, plastic,
What kind of chips do we have over there?
KAP something. Lots of crabs. These are Galathea crabs, so different than the ones we just collected. Amazing. Well, we want to know how to collect human debris. Use the tube arms.
We can only figure out a way for them to eat it.
Yeah, well, tube worms don't eat. They have no mouth.
Product placement.
Product placement, yeah. I don't think
Puzzle
so.
pieces.
Coca-Cola.
Yeah. Gods must be crazy.
Did somebody say Riftia?
Isn't that Riftia?
These are lamellobrachian.
Oh, okay.
Yeah. I was gonna say, let me know if you see a Riftia. That would be very exciting.
What's the difference?
Well, Riftias are bigger and fatter. Morphologically, they're different, but since I don't study tube worms, Greg will have to tell you what the big difference is. Let's go see. There's a big mound up here full of tube worms.
Yeah.
Look at this site. Magnificent. Jeez.
That's where we want to put the
This
marker.
is a lot of tube worms.
yeah, that's the spot.
That's the spot
This
for the marker.
is a lot of tube worms. Funny that this has never been seen before.
It's pretty cool. Like a
It
two-worm
is
egg.
very cool.
Some fish line going across it.
And the forms it's almost like the tube worms, like corals, are making their own reefs. They make
Yeah.
a structure that probably alters the currents, probably affects food supply. So let's just take some time and look at it. see what creatures are there. So we've got hermit crab and the crabs. We have galatheid crabs. Ooh, we have some like a bivalve just sitting there. Maybe an acesta. Is there a fish inside there? Is that what that is? Okay, well, I'm hearing that people want these snails. These snails we've seen before on the tube worms. Yeah, you got some already, but he wants more? We got 2 yesterday? Cool, okay. What are, there's little things, zoanthids maybe, on little Nidarian polyps running down the tubes. And lots of hydroids. A couple of kinds of shrimp. Where? Oh, at the top. Look at that. That's pretty unusual. Sea stars don't usually.
Tree. Right. Christmas sign, seasonal.
This does look like a Christmas tree. Pretty busy place.
Yeah.
So all the fuzzy brown are hydroids, which are colonial cnidarians. And we've done some shipboard experiments that suggest somehow hydroids can take up carbon from methane. So when we label methane, that label ends up in their tissues. So they may have symbiotic bacteria that can oxidize methane, or they're eating them out of the water. Oh, look at the fish.
Can we just stare at that for a second?
Maybe put a little BBC highlight button on. Yeah. Here. So basically, we're at a really large mound with quite a few tubeworm bushes, whatever we want to call them. This was found by mapping with the M3 earlier today, I believe. So it's a really nice combination of geologic tools combined with biologists who can come view and decide what we're looking at. We'll have to find a way to take some samples without too much disturbance here. This is one of the biggest edifices I've seen of tube worms. And that looks like a really pretty large old fish hanging out inside. Not sure what kind of fish that is. Maybe it's just a big rat tail. We're being asked why, in the chat, why one side of this edifice is more hairy than the other. So the hairy are the hydroids, and they may have greater access to methane in the water or currents. Whoa.
Wow.
Maybe we should have a contest to see who can count the tube worms.
There's another sea star at the bottom.
Oh, look at How many tube worms do we think are in this cluster?
Another swordfish.
Could AI tell us that answer?
Peter, you've been dropping an
Probably.
you've been dropping an observation for each individual tube worm, right?
So the hydroids are more at the top, fuzz, and it's much cleaner down below. So we have requests for the snails and the starfish. We have coral quivers on the vehicle. Would those be a good place to put the snails and the starfish?
Yeah, we can pop it. Yeah, depending on the size of the starfish.
And how soft this is.
This is
really mad.
Yeah. Yeah.
So I just brought up let's see. Just brought up a PIP which shows the M3 mapping that we did in the beginning of this dive. Jeff has worked tirelessly to map this, and here is the mound that we're currently on.
Yeah, this mound has a little mound on top of it, which we didn't know what it was when we mapped it this morning, but now it appears to be a giant tube worm ball sitting on top of it. It's a giant mound. I don't know.
We could call them bushes, we could
Big
call them
bushes.
clusters, or we could make up a new name.
Yeah. I was hopeful that the map that we made this morning was actually seeing the tube worm cluster, because
It is.
yeah.
Yeah, you predicted that.
Look at the big ones in the back of this shot too.
Well, I'm really glad you had time to come over to this thing. So cool.
The map is beautiful too.
Yeah.
Nice work on that.
So there's an interesting hole in the middle.
That's the M3 map. You can, Juliana's zooming out a little bit.
Sorry, you want to zoom out?
Ooh, could we zoom in on the orange? See what that is?
Oh my goodness.
What is that? Is that microbial mat?
How cool is that?
Okay, loggers. Put that in, please. Oh, the crabs are eating the bacteria.
Not yet, but it would still another 2 or 3
Was that
minutes
on the
until
big
I would
mound?
see a
Was that all still the big mound?
big mound, and then just poking and
Around,
sampling.
okay.
That was just like,
Yeah.
not surprised by that. This was from here to here was sitting in one switch.
So this looks like a tube worm nursery because there's little baby ones.
I'm telling you that.
You guys see the baby tube worms?
And it, oh, yeah. Look at those
That's the
little
first time I've seen really, really little ones. So maybe they start
It's a nursery.
in there. Can we like zoom in on the tiny ones? So over to the left a little bit, there's really little lamellobrachias. That's kind of interesting. There's a crab. So we have documentation. The crab really does look like he's eating the orange bacteria. Right? That is cool. Let's take that video for a little while. Just
this is the mission control, as Sophia introduced it. Doug's flying right now.
If anybody doubts the crabs eat bacteria, we've got documentation.
It's even using a plate.
Just keeping very steady camera view of this crab. So for me, this is my favorite room on the ship. The pilot's gonna sit there, they're gonna control the flight of the vehicle, we call it flying, you're not driving, you ROV. The co-pilot sits here, and they help out with navigation. They also run the manipulators, so those are the robotic arms that Sophia may have showed you an image of. And you control that with this controller, which is a small replica of the arms that move this. And the arm moves exactly the way you move.
I wonder
Close and
what
drop.
the bluish
A little bit ago,
on
we
that.
were push motors, so we were shoving tubes into the seafloor, pulling them out very carefully, putting them in tubes on the cell.
Those could be folliculi.
There's some other fun ways to sample. So if you pick up a rock, it's a sample. If you cut a piece of coal, it's a sample. If you grab a fish, it's a sample. So we can use the manipulators to grab things. We also have what's essentially some vacuum there on the sub, so we can essentially sample things.
Definitely some baby tube worms over there.
As well. But as you can see, this is a very busy space. There's a
The
lot
hungry
going on.
crab.
You also have to watch a variety of screens. You have 10 different camera options that you can cycle through to get an idea of where they are in space, where the arms are in space. There's no feedback, you can't hear anything, you can't feel anything. It's all based on sight. So we do all these camera views to create a three-dimensional image of what's going on in our heads. So we found this very cool website. It's probably turn back to get Doug in and keep doing our science down here. Anything you'd like me to add, so.
Perfect. Thank you so much.
Sure, all right. Thanks for taking a glimpse of the mission control, and you can always watch us on YouTube, Twitch, and Facebook.
Lisa, how possible is it to grab this crab?
Let's ask. I'm not sure that we're not in a, are we in a place to sample, or do we need to sit down if we wanted to take samples?
Where you want to sample?
Well, I was asked for the crab, and I know
The crab
with,
that's eating?
yeah.
Oh, man, that sounds terrible.
I also think it's which core refinery is it?
Well, no, not here. I've been asked for the starfish, the snails,
Yes.
and the crab. So I don't know whether it's necessary for us to sit down to collect those.
Yeah, you do.
Starfish is at the top, the snails were on the other side, and the crab is here.
There's a couple snails here as well. Those work for you.
Yes? Right. Yeah, the white coiled, sort of volcano-shaped snails. Yeah.
We suggest a heads up. The WhatsApp is saying they're happy without taking snails at this site.
Okay, I'm settled down.
Was that Javier said that? Because Javier is the one that asked for them.
It's, yeah.
You want another one?
She asked for them.
I think they want that crab, but you might be able to grab it with the net.
Yeah.
You want that in a bio box, that crab?
Could it go in a, yeah, but bio box is okay. Or a coral quiver, but, or a suction.
All right.
Whatever's-
Dealer's choice, whatever we can grab it with. Well, we have one shot to grab it.
Fit the suction.
I don't think so.
Let's do it. Biomass. All right.
This orange mat's really special because of the really tiny tube worms that are in here. There's also a bunch of really small ones to the right of the crab. Poor guy. His last supper. Poor gal.
Maybe.
Got it. Grab some supper.
You got some to go, buddy.
All right.
Would it Would it fit in a quiver, coral quiver, or do you have, you can't use them
Can't
because?
grab it now, we had to open it
You
prior.
had to have opened it, okay. Go ahead and put it in a bio box.
Which one do you want it?
Just whatever you tell us you're gonna use, it's fine.
4.
They're all empty.
Okay.
So I have conflicting information. Pachi says she wants snails and Eulogia says we don't need them. What's the final verdict? No. Okay. So the starfish is the next priority?
Yeah, we need the starfish. There's a small one, the white one.
While you're here and I got that out, might as well open up one of those coral quivers, because if we can grab the starfish, that'd probably be a good spot for it. Are you happy with that? Or would you rather it in a bio box?
You can go in the same bio box. You
Same
just,
bio box, okay.
did you just use 1B or 1A?
That was 4.
4B. Thank you.
Alright. For the starfish, probably gonna have to hover there, so. Get those stable things.
And I don't know if Greg wants any of these tube worms.
Always messes me up with the starboard port, starboard. I see the star. Hey.
This large tube worm edifice clearly has sort of sectors with different things in different places. It's interesting. The hydroids, which make them look fuzzy, are only on one side.
There's the starfish on the top.
They're going to it, I think. That starfish right there. Looks like the rat tails, or whatever fish that is, are hanging out inside only in one area also. I wonder if we should take a temperature measurement here. Scott, are you seeing a signal?
Big spike.
Big?
Yeah, the same size. It's going up and down a little bit, but it's about the same size as the biggest one we saw yesterday.
There's a sea cucumber. So big methane spike here.
Nope, slid down.
So it'd be interesting to know whether the asteroid is eating the hydroids or the tube worms or something else. They're predatory. It can, either, whatever's convenient. It can go in same place 4B. Yeah, just as long as the crab doesn't swim out. Thank you. Let's take a look at the sediment surface down right below us, the gray and black patches, just to see what's there. So Greg wanted long form tube worms. Do you see? I think the ones right in front of us might be long and the other ones are coiled. What do you think? The fuzzy ones that are hanging straight down.
Possibility, yeah.
So we were asked to collect some of those. There's a big question about whether they're different or the same type as the coiled ones. Let's just, since I asked you to go look at the sediment, we'll go do that first.
Is that what you want to do or not?
Yeah. Yeah, let's go look at the sediment first and then perhaps collect some tube worms from our upper left. I'd like to zoom in on the grayish patches and just see what that is. So what do you guys think we're looking at? Do you think this is folliculinid? It does look a little, does it look bluish to you?
A little bit, like on these more
Yeah, like
defined
right
pieces,
here.
It looks a little blue-grayish.
Right. Folliculinids are ciliates that form mats at hydrothermal vents and methane seeps. They have symbiotic methane oxidizers. So we're always on the lookout for them. They're probably different types. So maybe we could suction right where things look especially bluish.
Okay. Sure, I mean, I just have to reposition just a touch, but if you want, we can grab this suction sampler all apart.
And I mean, those black patches also could be some of them. I can't tell if they're black, green.
Which jar do you think you're gonna want that in?
Maybe 3 or 4. And before we actually sample, do you think we can, are we too far away to zoom in on those greenish?
As far as I can. And then
Yeah, there we go. Okay, that just looks like mud. So that's good. We won't want to sample those probably. They just look like sediment. So let's try for sort of the grayish bluish areas.
I am lined up with number 4, yes. You're welcome.
I love that that big old fish has a home inside the tube-worm cluster.
Settle down here, and you should have a good work there.
So yeah, I think it'll be, hopefully the tube worms won't come in and clog up the
Right.
thing. But they should be pretty well attached.
How deep do you want me to go? Just the surface blue?
Yeah, with, the upper centimeter.
You see right in the center of the screen, really where it's
It's
blue,
kinda
right where
right.
the worms kind of have. Yeah, right there. Alright, just some suction there.
I don't know what the texture is like. You could try going into it. Let's see. Yeah, there we go. Kind of muddy, huh?
That was a big chunk.
It's a really interesting rubbly kind of texture, but I'm just wondering if these are folliculinids, they've probably taken the mud and turned it into beady shapes. I don't know.
Would you like something anywhere else on the top there, This area
Let's take a
here?
look. Ooh, look at all the little baby tubes. Sure, grab some of that. Let's see what. Looks like there's very small vestimentiferans in there. They're still on there. Okay, that's probably good.
Good.
Yeah.
Alright. Are we gonna
let me see if,
Just hold on to the tube, it's not a big deal.
well, I think the next thing we're gonna wanna do is collect some of these long tube berms that are right there, so we can probably stow this section for the moment.
Just letting that hose clear a little bit, make sure everything's out there.
So that was number 4?
That was number 4, yes. Got to start with this then.
So we're more or less at the base of the mound, right, with this sample?
More or less, the base is just below us, I'd say we're probably maybe a third of the way up.
Let's see. Down to flank.
And where are you gonna want these tubes, or you want them in the bio box?
In a bio box, yeah.
Let's see if you can reach some of these. If not, I'll have to draw us in a bit.
So Greg's really interested in, he said long form, so the ones that are coming like straight down, yeah.
Draw us in a bit.
He'll be in here in a few minutes too, so.
Just watch that, but that's like some fishing line or something right there.
That does look like fishing line. It's a shame. We don't have a way to cut it, do we?
You can try cutting it with that blade, but I don't know. You can try cutting it.
The hydroids and all the filter feeders don't care whether it's a tube worm or a fishing line when they settle.
No, it's not gonna cut it. No, just let it go. That way we don't have a free end floating in the water.
I guess coral cutters and fishing line cutters are different.
They're a little bit different, yeah.
Oh.
So grip these as high up as possible, is that what you'd like? Going one at a time or just holding?
I guess the open ends are down, right? So the base must be up near the fish or something. I'm not so sure. We don't want to pull the whole bush down, so I'm a little worried about that.
I can grab one lower down and give it a tug and see.
Yeah, you could try that, see if what happens. Ooh, that's what I'm worried about. The whole thing will come off.
You see out right by the fishing line there, try that one. Cross above it. Yeah, just there now. Yeah, that one there, you just kind of
Well, that might work.
looks like it's flipping around the same.
Yeah, okay, let's,
We have to open those prior.
it's, okay.
You want more?
if possible.
Anyone that you see, Tyler, that might be accessible. You can see that one back there. You see them, both of those get a little peaked in.
I don't know if I think we need to try for the one, to be sure that they're not the curly ones.
Okay.
Want something over here.
All right.
So that was BioBucks 4A for that tour. Got you half of one.
Thank you.
You're trying for another. I assume this is what you're calling log form.
Yeah, it's so different to the other.
They do look different. So, core-wise, there is a reasonable background spot, sort of right 50 meters back on our track, or 25 meters back on the track that we're on. And we need at least, we need 4 background cores, 3 for Marlow and 1 for Puff.
Okay.
But there's still a lot more to look at this place. I think we barely touched it. Pretty impressive spot. Yeah. So you saw what I did. I tried what might be a folliculinid suction, and is there somebody home?
Look around.
Can we tell?
There's something there.
Yeah. Okay. Great. So back in the same place with the other B4A, just right to. What else did we get? So we got miscellaneous crabs, 1 whelk, 1 fish off the bacterial map, snailfish, I think, a liparrid. So that's no rocks yet, but we haven't been anywhere with rocks yet. So, and there, it doesn't look like there's any rocks around here, but we should make sure to get a good, I don't know, probably with a photogrammetry camera.
This looks like a good one to try.
This should be excellent for that. And I don't know how that works, if they just go back and forth over it or circle it or whatever. So the only waypoint we've been to so far is the orange map. That was most of the time.
Yeah.
And does somebody want an disc here? Well, maybe we should take a disc file from here so that Anna has a heavy duty, because there's a really strong methane signal here.
Okay. So, okay.
We've only done one.
Ooh, that's a long one. Let's see if we got some.
Ooh, that's a good one. Plus, there was somebody on it.
Oh, yeah.
Great.
I don't know how many you
Is
need.
that empty?
No,
That's
that's,
okay.
looks good.
Okay.
Well, 10 would be good. But
Okay, well then have them keep pulling.
yeah, we'll do it. Okay. Enjoy lunch. It's good. The chickpea curry is delicious. Oh, you
You can't leave till.
Hiya, Doug.
How are you today?
Very good. Great sampling yesterday.
Yeah? Good stuff?
Yeah, kept me up. That's fine. That's what I'm here for.
Greg, the sister is very close to the border between the 2 boxes. Just a heads up.
Don't let it come out.
Can we
Or
push
move.
it?
Push it back in? Yeah.
The sister is on the bio box 4B.
Yeah, we're
There's
gonna
several
push it.
things in there that should not come out.
I see. Okay.
Thank you guys so much.
Yeah.
Yeah.
Great work.
Doug's actually been noticing that. I should get more hours on
I should say goodbye. Bye, everybody out there. Get Greg
All
Rouse
right.
now.
Thanks, Lisa. Lisa's off to have lunch, and I'm Greg Rouse, and I'll be with you for the next few hours with the team. We're at the El Quisco Seep, off kind of close to Valparaiso if you look on a map of Chile. 420 meters of water. This is a feature we didn't visit yesterday. We were exploring yesterday, and the mapping team found this other mound that is pretty spectacular with lots of vestimentiferous tube worms, and that's what we're sampling now. You can see actually there's two kinds of these tube worms. Some have long tubes and others are curly and white. So yesterday we got some of the white little curly ones, and today we want to get some of the longer ones, and we'll see if they may be different or they're just morphs of the same thing. Got a bit of a changeover happening now. The ROV team work 12 till 12, so they're on 24-hour operations. We have two teams, and then they have shifts through those 12 hours. And then for the science team, we also have 4-hour watches, and lunchtime here is 11:30 to 12:30. So we switch out at 12 for, and the first watch started at 4 a.m. actually, 4 a.m., 8 a.m., 12 p.m., and we final watch is usually 4 p.m., and we tend to come up around 6 p.m. But we're a bit flexible, luckily, because we have 24 operations on the ROV. And on the watch today, I'm joined with Amanda and Fernanda and Peter, and they'll be talking to you through the afternoon as well. Hey Scott, what I'd like, please, is a few more of these. We've got these kind of curly little ones, which we've got some of those before, but we didn't get many of these longer ones. So if we could pull some individuals out. If you can grab a few, that would be great. If you can grab a handful, that'd get the job done.
All right. And into a white box?
Yes. We're going into bio box 4. 4B.
4B. Is that crab going to escape? Or is that squat lobster? We don't have names, we have pictures.
Yeah, it is a crab. Oh yeah, cool. That's a crab. I recognize it.
Have you seen our illustrations?
We have maps and logs of where the samples go, and people have their different styles.
I've been working on illustration, science illustrations. That is not one.
Yeah, a squat lobster would have 3 legs and 2 claws, and a real crab would have 4 legs and 2 claws.
We'll probably do a curvy tail as well.
Yeah, yeah, with a little tail. Yeah.
Get my head in the game.
All right. I'm here with Scott and Ben running the ROV, and Julianne's running the social.
Is that a good spot there? In there?
Yeah, that'll be fine. Yeah. I'm sure they're in there. They're pretty, the tubes taper, so even though they pull in when we grab the front, the tube, we should get them, even though the tubes are long, they can't go too far back. Yeah, as far back in as you can.
So
These
there's a
tube-
chance that breaking these off shallowly, you'll miss the animal entirely because it retracts back into it.
If we grab just the tip, yeah. They can pull in a fair way, but then the tube tapers and they can't get down far enough. The worms are actually very long. They could be maybe a meter long, but they taper to very thin back ends. And last night, for instance, we didn't get any complete animals at all.
How big of this bush do you want?
That's a good bunch. I like it. So why do I want 10? Well, it gives us an idea of the genetic variability in the population. So we'll sequence the DNA of these. So one is not enough, and 10 would be a good number. And unfortunately, it's hard to pick them one by one, but with this sample, we should be able to get enough information. Oh, that's great, Ben. And you can see how long the tubes are going back down there, and that's why I'm interested in comparing these with those short little curly ones. Are they the same? A grab of that would be great. Thanks.
Hold on, we're doing a picture. How close up do you want?
That's good enough. It gives us a scale. Thanks.
Ready for draw out then? Watch out. Good.
Oh, they're going in A.
We're gonna go to A. Who's A? To the left. Okay. This right here.
That's BB4A.
Yeah, it's still the same sample. It had started already. So these tube worms are called lamellobrachia. They were first named in 1969 from just near the San Diego, California, Mexico border offshore, and they were discovered by a Navy submarine, a submersible, experimental submersible, and that was kind of a big discovery. Subsequently, they were found at hydrothermal vents. So it was a little methane seep off California, then at hydrothermal vents, the very famous ones called Riftia that you might have seen. Any movies from hydrothermal vents you would have seen. And so these vestimentiferan tube worms, lamellobrachia, Riftia, a few others, live at either methane seeps or hydrothermal vents. Sometimes they've been found in strange places like well falls. Did we get, well, we don't know. Can we get the laser on, and we'll just get a scale on the tubes? And if any of these long ones are out, we can zoom in and get a nice close-up. So we'll just get some close-ups of these long form ones and compare them with the curly Q ones. We could look at the ones that were over here, were out, were probably good enough. A little bit further over. Yeah, up there. Those are good. Yeah, that's great. So here you can see the feathery crown that attaches to then a long wormy body. The body, part of it, is full of bacteria, or about 20% of it's bacteria. And the red is from hemoglobin, blood sort of similar to ours, but different in that it can also bind hydrogen sulfide as well as oxygen. And so these worms are farming bacteria in their bodies. And that's why they can live and thrive at methane seeps and hydrothermal vents, where the main food source for many microbes and the animals indirectly is that hydrogen sulfide.
What are all these shrimp-looking things drifting around the waterfall?
A lot of them are mysids. They're a kind of crustacean. They're often quite small. And yeah, I think most of those are mysids. And then there are real shrimp that we've seen more hanging on the bottom. But mysids are usually pelagic. Some of these might be shrimp we've disturbed that are up off the bottom. Mysid spelled M-Y-S-I-D. We've got a little squat lobster poking its orange claws up there. The bushy things you can see are mostly hydroids, the polyps of hydroids. So hydroids bud off little medusae that end up as pelagic little jellies. There's another squat lobster. And some little octocorals, the little polyps you can see there. Transparent. I saw a sea star, Scott. I'm not sure if we sampled it. Yeah, there's definitely mysid swimming, and the sea star is here. They did sample a sea star earlier, so I'm going to assume it's the same one. Anyone from the previous? No. No one was here. Yeah, we don't know. We could take it just in case. Ben, could you grab that little sea star, please?
Yes, sir. What do you want with
It can go in BioBox B4.
Dead center.
How tall is this feature, Scott?
Can we? Definitely over a meter.
Yeah.
Maybe even 2.
The multibeam showed about 4.
There you go.
Wow.
3 to 4.
This feature meaning the mound or the?
I'm just looking at, that.
Which feature? Oh, that feature.
Yeah.
Sorry, I thought you meant this.
Yeah, Sebastian is not to scale in that map, you see, but it looked like about 4 meters.
Okay.
That's huge.
Yeah, this looks like something we'd like to use photogrammetry on to make a 3D model. So there's a special camera mounted on the ROV for doing that.
Is the M3 image picking up the 2 worms as well, or is that just the underlying carbonates?
It's picking up the 2 worms. Yeah, it's just the mass.
Just kind of
It'll return on pretty much any solid object. We were seeing squid in that.
Oh, I see. Okay, so it's not like density.
No.
Oh, look at that. So good. Whoopsie.
We'll grab it, it's fine.
That was just to make you guys all excited.
He has an arm up for you.
When Ben's done with this, there was the snail crawl. We'll come back for a close-up. We've already collected the snail. We didn't get a good video. So the little arms have silicon tips so that Ben can pick that up without damaging it. B, please.
Starts out that's as far down as it can go.
Great. Thanks, Ben. And I think we'll not open that one again. So we collected in these tube bushes yesterday some very beautiful snails. And I took some photos of them this morning, but it's always nice to get a good live image of it crawling around. So these are going to end up in the National Museum in Santiago, Chile, and we'll get a nice video of this one that's crawling along the mud in front of us. Yeah, this little guy.
See that guy?
This one had two cute little eyes. Even though we're at 400 meters where it's completely dark, sometimes the creatures that we find down here still have eyes. Shell's nicely iridescent, leaving a trail, but we saw a lot of them up in the bushes.
Where did he come from? Way up there.
Yeah.
Took days.
So we won't sample this one, but we'll refer it to. Can we do what kind of frame grab? Not a sample frame grab. Yeah, just a BBC. Maybe do a BBC frame grab. Thanks. Okay, thanks, Scott. What I'd like to do is some sloping, please, up in the tubes.
Sloping?
Sloping, suction sampling.
You want to get a little closer to that? Which part do you want to slope?
Where you were grabbing them before. Can you do
Where
that?
the lasers are up in there?
In there? So underneath all of these worms is probably carbonate rock. And the worms are, in a sense, almost being like a coral reef, and they create all this extra habitat for other creatures to live in. So if we can sample those with a suction sampler, we can find lots of small animals that we otherwise wouldn't see. And we'll Mostly it'll be sediment, but living in amongst the sediment will be all sorts of little creatures. So we sort those out under a microscope and keep them cool, take good photos of them, and then preserve them for further study. So, Julianne, can you show some of the microscope pictures if we give them to you
Sure.
on the feed? Yeah. So we might show some of those tomorrow.
Okay, and you said you wanted number 2?
Yes, please. So we'll just poke in around.
Can you use your stick up, Greg? Just anywhere?
Yes, thanks, Ben. Just in deep in amongst the tubes would be great. You got me some good things yesterday with the slope like this. So we'll see if we can get some more. Yeah, we can see.
What section sample jar is this going into?
Number 2. Looks like there's 3 different kinds of squat lobsters here from what we sampled yesterday. Yeah, that's good. Thanks, Ben. That's great. Just trying to tell how full the chamber is. We want the creatures to be able to live on after this, but they should be fine. Their tubes are very robust. Okay, I think that's great, Ben. Thank you. Not right now.
Okay, so we can stop.
Yes, please. So this seep was first noticed by some catches that fishermen made with their lines, and we've seen a lot of evidence of lines here. And that led to some initial survey, not with ROV, but they managed to then get some of the clams that live here. We've seen a lot of the shells. And Elena Krylova and Javier Solanas, one of our Chilean collaborators, named that species, and it's called Austrogena nerudae, after Pablo Neruda. And we've yet to see them alive. We've seen lots of shells, so one of the things we'd like to find today are those clams. What we could do here, though, is take a Niskin. This is for Hannah, for various things. High methane. It's a good methane reading here, Scott, right?
Yeah, it's heterogeneous, so there have been high and baseline all here. But we have high readings.
Okay.
This would be bottle number 2, should be.
Number 2. There we go. So that's a 5-litre bottle of water, and that will be filtered on recovery. Could be used for eDNA, could be used for looking at plastics. Could be used by the microbiologists.
That was 2.
Yeah. Scott, could we just look down here where those clam shells were and just see if anybody's alive down there? So I see that clam there. I think it's dead. Where are the live ones? I wonder if they're in the sediment. Yeah, we could do the a scoop here of sediment. Ben, if you feel like running the scoop through the surface of this sort of muddy area right there. Maybe we'll get a clam shell. And then this will go in one of the bio boxes. So this will be a sediment scoop active.
middle scoop. Is that what you mean?
We don't have a scoop with a bag?
No, no, what's tooth? Can you zoom in? This side? Yeah. You see a scoop.
Did they get rid of the scoops because we're recovering the quivers?
Scotty, I think they're on top of, at least before we dove, they were zip tied on top of one of the bio boxes.
Okay.
Oh.
About to ask the, all right, porching out. Amy, moving. Ah, there it is. Okay. Thank you.
I got you.
Yeah. Okay.
So those metal scoops have canvas bags attached to them. And to catch the sediment and then keep it reasonably secure while we recover the ROV. We'll put it in one of the bio boxes and then sieve it once it gets to the surface. Gives us another idea of what's living here apart from the sloping.
Inches away. Just clears.
So there's a shrimp swimming in the frame, and the smaller creatures that are zooming around though are mysids. Looks like this is scoop one, or otherwise call it long handle.
Really clear so far.
There might be carbonate directly under this, or it could be reasonably deep. We don't want to go too deep though. Yes, that's great. Thank you.
Just one inch.
Yeah, the top inch, please. If we see any clams, oh, it's pretty hard.
Want me to grab it so you can re-grab it?
It's just rock right underneath it. Yeah. I think over here, when you look at the, whether there it's maybe. Is that other scoop with the silicon handle easier to use? We should make sure we use that. Well, right. If we try around here, might have better luck. How far offshore are we, Julian? Oh, Sophia, how far offshore are we? 10 miles? So we're about 15 kilometers off El Quisco, holiday town for many people from Santiago. It's just south of Valparaiso. And this seep's been known for about 20 years because of fishermen catching different kinds of sea creatures with their lines and then giving them to scientists. So that indirectly led us to coming here. But now we've been able to visit with Sebastian, do wonderful mapping. And we were very surprised yesterday at how many tube worms were here. Oh, that's great. That's good. Thanks, Ben. You can see bubbles coming up. Lots of clam bits. Hopefully we got a real clam.
this is going to.
What's the any of the bio boxes that are so B1, BB2, BB3, whatever's easiest for you. Do you think we'd fit two?
Let's see what happens.
Okay. The tubes, you could keep the tubes too.
Yeah.
Oh,
Sure.
okay. So there you see the other style of scoop with the silicon handle. We'll use that in a little while.
Can you check the camera? It is, yeah, all the way. Zoomed all the way out.
Ask Fernando or do you want to come ahead and speak? Do you want to say talk to Spanish?
Okay.
Introduce the dial. Yeah. Yep. And the sampling for the sediment. Yeah.
Buenas tardes, les habla Eulogio Soto, jefe científico de la expedición, biólogo marino de la Universidad del Paraíso. Nos encontramos en este momento estudiando, explorando un Seep, que son ecosistemas quimiosintéticos. También se le llaman ecosistemas de filtraciones de metano. Y hemos podido encontrar un ecosistema con bastante abundancia de tubos, de poliquetos, de nombre lamelibrachia, los gusanos cipoclínidos, que son los que se ven en la imagen. A través de el robot Sebastian, en este momento la imagen muestra cómo a través de una bolsa o un saco es posible recolectar sedimento en condiciones con bajo contenido de oxígeno, que nos interesa para poder conocer la fauna de pequeños invertebrados que viven sobre o enterrados en el sedimento. Entonces, a través de el uso del brazo mecánico del ROV, podemos utilizar esta bolsa o saco para poder obtener sedimento.
So that was Eulogio Soto, a professor from Valparaiso, and Eulogio's our chief scientist on board this leg. We'll be on for a few more weeks here at El Quisco for another few days, or a remote canyon nearby called San Antonio. Then we're going to head south to Concepcion and explore some more seeps and canyons down there. and then continue further south, and ultimately we'll finish the cruise in Puerto Montt in early November. So I hope you can join us for a lot of those dives. Hello, could you find Pedro or Dan from the BBC? Or WhatsApp me? Are they on the WhatsApp?
So Cam is your friend. Can you zoom? On what?
We're just stowing the scoop away so the biobox can close properly. The handle creates a bit of a problem. Yeah,
Close it. Hopefully it goes the correct way.
Buenas tardes, nuevamente les habla Eulogio Soto de la Universidad del Paraíso. Comentar que durante este dive, durante esta inmersión que está siendo liderada en la sala de control por Greg Rose del Instituto Oceanográfico Scripps de la Universidad de California, pero también nos acompañan otros científicos de la Universidad de Boston, como Amanda Semler y Erin Freitz. También se encuentran en la sala de control del ROV Scott Vina y Fernanda Orrego, quien realiza estudios de zooplancton y también es bióloga marina de la Universidad de Valparaíso. También se encuentra con nosotros Lucía Villar, geofísica de la Universidad de Chile. Comentar también que en esta expedición se encuentran a bordo 2 miembros del staff de la BBC de Londres, camarógrafos que también han colocado sus equipos técnicos, sus cámaras, para realizar estas pioneras filmaciones de filtraciones de metano en aguas chilenas. Es importante destacar que nunca se habían filmado con este tipo de resolución ni con estos equipos este tipo de ecosistemas en las aguas chilenas. Por lo tanto, durante la expedición también tendremos el apoyo de este equipo de camarógrafos de la BBC, quienes posteriormente realizarán un documental como parte de Blue Planet 4 de la serie de Deep Seas. Actualmente, ahora pueden ver cómo el ROV ha subido un poco desde el fondo y se ven estas importantes agregaciones de krill sobre la filtración de metano, y nos encontraremos realizando observaciones y recolecciones de organismos en diferentes lugares. Recordar que estos ecosistemas son muy dinámicos, son muy variables. Para el ojo común podrían lucir como ecosistemas muertos o pobres, pero la verdad es que pasan también por diferentes etapas de sucesión ecológica, en donde hay una combinación de organismos muertos, pero también nos hemos encontrado con organismos juveniles y organismos en estado adulto. En algunos casos encontramos mantos microbianos que pueden ser de color blanco, o incluso hoy día encontramos también de color naranjo. Sobre ellos hemos podido ver cangrejos, gastrópodos, caracoles marinos, estos tubos de estas agregaciones bastante exuberantes de poliquetos. Hemos visto peces, diferentes tipos de camarones, Y también recordar, así como lo comprobamos ayer, que estamos en un ecosistema de alta productividad donde también coexiste por ejemplo, el calamar chileno o jibia, y también hemos podido encontrar tiburones y otros peces. Saludamos entonces a todos quienes nos están viendo y escuchando desde la plataforma de streaming de Schmidt Ocean Institute. Recordar que estamos en el barco Falcon 2 de este instituto norteamericano, realizando exploraciones inéditas de ecosistemas únicos, nunca antes explorados frente a la costa de Chile. Esta expedición comenzó el día 11 de octubre desde Valparaíso. Los días anteriores estuvimos visitando el cañón del río Limarí, donde también pudimos obtener registros de ese cañón, conocer la vida bajo el mar. Posteriormente, después de estudiar este ecosistema acá en el Quisco, iremos hacia el cañón del Maipo, del río Maipo, y posteriormente iremos a estudiar el cañón y los ecosistemas de filtraciones de metano en la zona de Concepción, para finalmente terminar la expedición el día 4 de noviembre, llegando a Puerto Montt. Después en Puerto Montt el crucero continúa en una segunda etapa. Hay recambio de personal científico. Sin embargo, se mantienen los mismos objetivos, que es explorar ecosistemas quimiosintéticos. Aquí nos interesa ir a la zona de la triple unión de placas. Esto está en la península de Taitao, en donde, de acuerdo a las evidencias, habrían no solamente filtraciones de metano, sino que también ojalá podamos por primera vez filmar una chimenea hidrotermal activa en ese lugar. Seguiremos en esa segunda etapa explorando el cañón del río Simpson en la Patagonia, y hacia el sur de la península de Taitao, después de estar estudiando la zona de la Triple Unión, explorar y caracterizar 3 cañones submarinos más que nunca han sido mapeados. Hay muy poca información batimétrica de ellos. También hay muy poca información, casi nula, de comunidades biológicas de profundidad, para que finalmente terminar la expedición en Punta Arenas durante la primera semana de diciembre. También se encuentran a bordo otros científicos de otras instituciones, por mencionar algunos. Se encuentra Marcela Cornejo, de la Pontificia Universidad Católica de Valparaíso, realizando estudios en la columna de agua, determinando y analizando los nutrientes, los gases. También se encuentra el oceanógrafo físico, el doctor Manuel Castillo, de la Universidad de Valparaíso, quien realiza estudios principalmente de oceanografía física de corrientes, determinando datos y procesando datos con diferentes equipos. También están otros científicos realizando estudios con sedimentos de la Universidad de Lafayette, algunos también de la Universidad de Nord en Noruega, estudiando el ADN ambiental. Y también mencionar que existen otros colegas de otras naciones que están realizando estudios con comunidades de fondo marino, estudios microbiológicos del agua, del sedimento, pero también mucha caracterización del fondo marino. Recordar que uno de los objetivos principales del Schmidt Ocean Institute es mapear, es decir, obtener mapas detallados del fondo marino, sobre todo estos importantes ecosistemas, como son las filtraciones de metano y los cañones submarinos. Entonces esto nos ha permitido generar muy completas descripciones del fondo marino, lo cual también es una información muy valiosa para la comunidad científica de Chile.
So this is
Okay, that was Oilogio Soto, our chief scientist. We are just sitting above this feature. This is the tallest feature we found in the mapping. And so it's a large carbonate mound covered with tube worms. And we're sitting up here because we have on board a sensor called SAGE. Do you want to talk about it? Might have Scott talk to you about what he's doing, why he wants this.
Cool. So yeah, SAGE is our methane sensor on board. It's out of the Chemical Sensors Lab at Woods Hole Oceanographic Institution. And it is a fiber laser-based methane sensor. So we're drawing gas across the membrane and into a fiber where we can then measure concentration. While this lets us measure really, really small quantities of gas, like the amount of dissolved methane in seawater, it does mean that there's kind of a response time that it takes for this gas to get across the membrane through the fiber. So to get a good reading, we're currently sitting on top of the mound for a little bit to see what we see how high it gets, and get kind of a good integrated reading from the very top of this big tube worm buildup, where we have been getting some large methane hits as we've been flying around and sampling. So it's giving us a single kind of point measurement for this spot.
All right, thanks very much, Scott. Good to go? All right, we'll have a look at the mound a bit more now, just to get a sense of how it looks, and we might sample a little more, but also we're interested in maybe making a 3D map of it. We've got a special camera mounted on the ROV. Courtesy of the BBC, that'll allow us to do a 3D map. We could do it now.
Okay. We got petrifications to expert the camera.
Yeah. Just all 10. Yeah. Within half. Look, yeah. And then do the map. Yeah. We go. Fly around so you get familiar with what it looks like. And then when he comes back, do you have a feed of his camera? That give a
somewhere. The photogrammetry camera. Do you know where that? Where the video is uploaded.
He said he had pictures in the feed of it.
What was that?
Where the photogrammetry camera can be displayed.
I can try something. Would you like
So you can see the long straggly tubes there, the tightly coiled tube worms there that are white, and then a lot of little white fragments, and those are the fragments of the clams. Austrogena nerudae. So these worms and that clam actually have symbiotic bacteria living in their bodies. The worms are all pulling in. Probably not because they see us. It's not the light, but they might feel the vibrations or water ripples from the submersible. The lasers you can see, the green dots there, are 10 centimeters apart. So this feature's several meters high, quite a few meters across.
Watch them all retreat.
Yeah.
It's like a little light show.
This is reminiscent of seeps I've seen much deeper at 1,800, 2,000 meters. So to see one at such a shallow depth of 400 meters is really a surprise. Off California, we have seeps at this kind of depth, but we don't see tube worms like this. Lots and lots of dead shells. All the things you can see swimming there in the foreground are mysid crustaceans, so they're not shrimps, it's another group. They're planktonic, feeding on other small plankton. Could you slurp on the fly, or is it better to slurp sitting? Do you want to do another slurp in this bush while we wait for the camera? And this could go in box tube 3, please. So we're going to do another slurp here in the bush of tube worms. And we'll get some of the little creatures living in amongst the tubes. Great. So this will be suction sample 3.
Suction sample 3.
sections on it.
And Ben, you can
On the bottom again?
go in amongst, but also see these white. Got 'em. Get some of those. And then push it right into the bush would be great. So you can see lots of sediment is in there, and probably quite a lot of microbial activity, and lots of little worms, small mollusks, crustaceans will be living there that we'll want to find to properly document this site. Yeah, that's great.
So there's no down there below you. There's no one down there.
Yeah, this is very good. Getting ready?
Just try and get the feed up on the screen so that RV pilots can monitor it while they fly.
Okay. All right, we'll do a little more here, and then we're going to try a photogrammetry run on this mound.
First thing obviously is take it super slow. So you want to avoid, the most important thing is to avoid kicking up sediment because that could affect the image to get better resolution. You want to kind of paint the feature, so look at the map there.
Okay, Ben, I think we're good.
The best thing to do is kind of ascend feature.
Thank you.
Descend the feature like that. We want to avoid orbiting. Okay. So I know this is easier said than done, but it's kind of painting the feature like that, up and down, zigzag the whole way around, and just slowly work your way around it.
Okay, so keep on the same plane and just go. Yeah.
So this is a different camera. We won't be able to share what we're doing with you. You'll still see the normal view. But we're going to try and fly over the mound at a fixed distance. We'll sweep backwards and forwards. And that should allow a 3D model to be made, and which will give us a great idea of the overall shape of the feature and a visually spectacular summary in just one model.
Do this kind of flat out dish.
They're just planning the way they're going to fly.
I probably start somewhere where you have a good reference from where you started, so that you can previously you come back to at the end.
Okay. I was gonna once he steps this, so I was gonna put the lasers as a reference over here, so my cameras are facing flat out, and my lasers down here will be my reference.
Okay, so is that cool? I think we can leave the lasers on.
Well, the lasers will be right.
Yeah, but you can set the camera level using the lasers. Okay. Turn the laser up, that's right.
Yeah.
But it's to find something at the base which acts as a user reference point.
Better not to have the lasers on, because the software
Then
might
you'll
grab
have
the lasers.
green dots that
The camera uses the same image.
Yeah. Unfortunately, these mysids might make trouble, but let's see what happens.
Yeah, it's so rich around here, isn't it?
Is it
stuck in the water.
How much light do you need for this?
What have you got on? Is that,
Yeah. The mysids are attracted to the camera, the light, sorry, of the camera.
we want it well illuminated, but I think anything we can do to try and reduce there's downward facing lights that are attracting all this stuff.
Yeah, that's what I'm getting at. So muds are off now.
The mysids are being attracted to the light.
To the light.
So if we can have wherever the camera is shooting from,
Yeah.
not have mysids attracted to that.
Right.
Yeah.
The other thing I've done before is turn on other lights, like the bullet light, leave that on so they go up there.
Okay. Yeah.
Half light on so they're back there.
Yeah.
And right now, I mean, that's the only light we had on. But we could do
We're
this.
just playing with the lights. We're trying to figure out the best way to image the mound for the photogrammetry camera.
Nothing.
This is a bit experimental. I mean, we can use footage like you see from the 4K, and we've built models before, but this is a different system to try and should give us much better models. The key is sort of flying at a fixed distance above and then backwards and forwards so that the software can properly reference fixed points that it discovers in the images and then build the model. It's getting very busy in here, so sorry if there's a bit of noise.
So you see the screen up there is the view from the first camera.
Okay.
It's basically the same view, it's exactly the same angle as the sit cam, but the important thing is that once we set the sit cam angle, we don't touch it. So you're at the base, now when you want to go down to the base of the feature and find something that's going to recognise the
All right. Just getting set up. You might have seen photogrammetry models of things like hydrothermal vents, but this could well be the first 3D model of a seep, and certainly it's a spectacular one to start with. We're going to be visiting seeps further south, so it's good to practice now.
So this is really close to this particular bush, if you will.
Yeah.
But as soon as I get above this, the next one is way off in the distance. That's like meters away.
This is all part of the same feature. So at the moment you're kind of on what we call the seafloor, right? And everything else is structure.
Yeah.
So I think this would be the best. It doesn't matter if the next feature is a bit further behind. It's just important that this is the feature here, right? So we're starting here, climbing out the feature. Rather than doing
Sorry, they're still troubleshooting the plan.
So it's okay when I come up above this to step in forward.
Yeah, the point about keeping the same distance, just to do with detail, because the colour changes so much underwater. Sometimes the sort of quite struggle to resolve all the images. There's massive differences in colour. White light.
That's a bug in our software.
So you can see we've got the image up there.
So which, oh, there it is. Okay.
We just adjust the signal and tell me when you're happy with the view. Is that fine?
Nothing.
Maybe that one. We just need to make sure we have
just
So that's, so you're inverted.
Yes.
Okay.
Oh
yeah.
shit.
So that's gonna happen.
Sorry.
See, I think that looks good.
Okay.
Yeah.
Is
Is it meant to be inverted? Or is that?
Yeah, it's meant to. Okay, so that comes out. Okay.
If I had like
Sophia, is it possible to share that feed online? Of the.
I'm sharing it with Tom in just a second.
Uh-huh.
As for whether he wants it on YouTube or not, just let me know.
Okay. So we have another perspective that is the photogrammetry cam, which is kind of cool.
So me sitting on bottom is turning up some mud, so we're gonna lift off a little bit. So you're carbon facing.
east south. So we think start.
Ah, those mysids are a pain.
That's how heavy it just turned out. More light, that's how light.
Well, they're attracted, we've pulled up from where the light was into where they are. So now they'll attract up to the light. I don't know how to avoid them.
It's more squid.
Hmm.
No, it's back to be a bit higher. So there was 3.6 discussed on 7. If you want to you can go to it, just walk inside, you can start trying what things that's not like that. This is really like that's where we start moving forward, where that is. Yep.
Do we want to square this up? Does that matter?
I think ideally you'd have more feature if you look at the calculation there. Feature of interest.
Oh,
It's-
yeah.
It's running through the centre because it's not fish eye. So now we're missing this due east.
Yeah.
Great.
We can call this a start point. So just go?
Yes. Just nice and slow session.
Hopefully as you get going, the mysids will be left behind.
Blend some of them in the rotors.
Yeah. And there you can see on the feed the striking difference between the what I'm calling the long form tube worms and the covered in hydroids and other things like forams, and the white form. Could be just the white form growing a lot faster. They're the same. Humboldt squid in the background there.
Was that slow enough? seemed like we lost a little light at the end there. So I can get back down a little bit. Reverse pass back to the slate. Give it a size of the feature. Paths are equivalent to an eighth of the size of that circle. Using Saturdays, like heading on 9 to March. That way. Yeah, that's easy to do, but we can kind of approximate that. But thanks. Otherwise, you can just come back down the way I came, readjust, and then go up. Okay.
So if you've just tuned in, we're about 15 kilometers offshore from El Quisco, a town just near Valparaiso in central Chile. And we're at 417 meters at a carbonate mound with active methane, seepage, and a lot of animals and microbes and wonderful life that we're trying to document here on our second dive at this site. And right now we're just making a 3D map of this place before we go and do some more sampling.
How did the methane end up, Scott?
Pretty high.
We stopped before above the mound just to see how much methane was ending up in the water.
So something that's interesting, Greg, is over the mound was actually, there was a spike, but it was less than when we were sitting trying to think about the photogrammetry up against that big bush of white worms. That was almost double the methane concentrations above the mound. So it'll be interesting to try and tease apart the methane data as we've moved all around this mound, because they're very heterogeneous.
Right, that's interesting. And of course, one of the issues about methane is when it gets into the atmosphere, it's a potent greenhouse gas. And the organisms living down here, of course, the things that are doing the prime moving and breaking down the methane and stopping it getting into the atmosphere are a consortium of archaea and bacteria. And they're the drivers of this ecosystem. And potentially saving a lot of gas getting up into the atmosphere.
So, rotate my head or rotate it?
The action of those microbes leads to the formation of calcium carbonate, the rock that we'll be sampling shortly, and also hydrogen sulfide gets released, and that then allows other microbes, bacteria, to live, and those bacteria are used by the tube worms you can see in front of you. So they don't have a mouth or a gut, they're just living off bacteria they farm in their bodies. As the same with the little clams, the white hash you can see all around. There's a bivalve that has bacteria living in its body as well. And then the crabs prey on them and all sorts of other things, so you get the complex ecosystem formed, ultimately all feeding off methane. So we're just doing a few passes over this mound, trying to get enough data to make a model. The little mysid crustaceans might create some issues, but we'll see. Looks like the worms are kind of used to the ROV. They're not pulling in as much as they were. Wow, striking difference there between the little coily lamellibranchia and these long, long ones strung out across the seafloor. So we're just about back at the base of the mound. We'll swing around a bit, go back up. So while we're continuing to map, I might have one of the other science party talk about their work. Hey Amanda, do you want to talk about what your team's up to here?
Hi everyone, my name's Amanda. I'm a PhD student. I just finished at Stanford. And I'm here working with a team from Boston University. We're looking at some of the microbes that are present at these sites. They're really special environments in the deep sea. There's a lot of carbon, there's a lot of hydrogen, and there's a lot of sulfate. And all of these nutrients together can be used by these organisms. They help feed some of these communities that you're seeing on the surface, some of these macrofauna. One of the things I'm not studying here, but I was particularly interested in during my PhD is how nitrogen is kind of produced in these environments. Methane is a hydrocarbon, so it has a lot of hydrogen and it has a lot of carbon, but it doesn't have a lot of nitrogen. And so one of the things many of the microbes in this system do is they take the nitrogen gas that's in the surrounding environment and they can turn that into forms of nitrogen that are more usable to the rest of the community. It's a really special ability because it's really, really hard to break the bond between two nitrogen atoms and nitrogen gas. And that's kind of another thing that helps these environments thrive. If the nitrogen was not being made by these microbes, maybe all of this life wouldn't be here.
Thanks, Amanda. And in a little while, we'll go and do some push coring, and we'll have Amanda talk a bit more about that. Her favourite.
Everyone loves the push cores.
I don't know.
It's a fascinating.
Some people more than others. Push coring can be frustrating. Luckily, the crew's already done most of the push coring today. They've done 11, so we've got 5 more to do. We definitely need to pick up some rocks. I haven't seen many corals today. We'd like to sample a couple of corals. And we could slurp some more creatures if we find any. There you see a little orange microbial mat. Can you make any comment about why the orange colour, Amanda?
I cannot.
Neither can I. We sampled the push cores in an orange mat earlier today, if you were watching.
Wow.
Oh, that's quite an image. Humboldt squid. Dacitacus gygas. Famous predator along low oxygen margins in the eastern Pacific. Although the oxygen isn't too bad here, we've been through the oxygen minimum zone on the way down, so it's quite a shallow oxygen minimum zone relative to further north in the Pacific, where at this depth in 400 meters we'd be right in the middle of it, but here the oxygen level is not too bad. Hence, we can see a lot of life. So we're continuing to go up and down as we rotate around this feature. And Scott's doing a great job just keeping a fixed distance away from it, going up and down, trying to keep the even lighting. And hopefully we'll get an amazing model of this. Quisco does have some marine protected areas associated with it, but where we are is not one of them. And we've seen a lot of evidence of fishing here, and a lot of the data we'll gather on this cruise will be made available with our Chilean collaborators and may lead to some action to do with this site. Imagine what a trawl net might do running over this mound. How's our progress? We've got good overlap?
So the starting point.
Oh, yeah, okay.
Back over, that's fine there. Close the circle.
So can we call it quits?
Yeah.
Okay, because I think we want to collect that.
Sweet. Perfect. Great.
We're right on front of a beautiful basket star, which we haven't sampled yet, and that would be a good one to get. Ben's back in the chair, ready to pick that up. I think that'd fit in a coral quiver pretty easily. So now the guys will go and process that data, and hopefully we'll see a fantastic model. So basket stars are a kind of brittle star, but the arms, they have 5 arms, branch. In this case, it's kind of closed up. The arms aren't really expanded. And so you can really get a sense of the central disc there with... They're just fixing up the lighting now that we had changed for the photogrammetry.
Now go for it. We need to get our lights back and everything back to where we want. So let's take a moment to do that.
This just in, apparently the orange color of that bacterial mat is caused by a heme protein that those bacteria have. And that protein allows them to reduce nitrate. So I'm assuming if we took a core in that place, we might see a lot of nitrate, a lot of nitrogen.
Oh, cool. Thanks, Amanda. Hopefully this morning's scores will be useful for you for that.
Yeah, I think we're gonna
sample that.
Yeah, clip it, put it in the quiver.
And we got that this morning, I think.
Yeah. Thanks, guys. Yeah, so you want this whole carotenoid?
It's a basket star.
Sorry,
Yeah,
basket
yeah.
star.
We'll take that into a quiver, please. I think it'll fit.
Okay.
Yeah.
Squat lobsters and crabs there together. Or both squat lobsters. 2 different things.
Oh, he's eating. Carrots. Yeah.
Yeah, it's 2 different kinds of squat lobsters there. That one near us has managed to grab a shrimp and is eating it. The pilots are always looking for predation events. We did catch some of these shrimps, and the taxonomist will figure out what it is. The lights we have off the sub probably confuse a lot of the animals, allowing predators to often make easy prey.
I was not expecting that when I zoomed in.
Oh, it's Sophia. Sophia chose that. Okay. So- So these little squat lobsters are commonly found at Seeps. They're a very diverse deep sea group. And some are particularly associated with methane seeps. We found 3 different species last night in the samples. But, we'll be checking out the DNA and maybe loaning them to other experts to figure out really what they are. Oh. All right, let's get us a basket star.
Thanks, guys.
So they're just next to the suction sampling hose. You can see 6 bottles with caps on, and those are what we call coral quivers, and that should fit the basket star pretty easily. Otherwise we'll be putting other animals in it like corals.
Looks like everything is open according to ordinance.
Yeah, any of those, whatever you prefer. Can we get a nice grab just before you grab it, Ben? With the light a bit better balance, please. Yeah, that's great. Thank you. Look at that. So the mouth is on the other side from us, so that's the top. So we can't really see where the food goes. Those arms, when they unbranch, have sticky. Well, they're not sticky. They're kind of spiky to grab hold of plankton. Right now, it's not able to feed until it spreads those arms out.
Ooh.
All right, that's a great shot.
So that's just lowers right now and muds.
Nice. Okay, please take it. Thanks, Ben. Yeah, that should work. Yeah, that's pretty strong. It shouldn't break. probably curled around that tube. Maybe if you grab the tube, might.
You want to grab it too?
Maybe grab the tube. Yeah.
This is like trying to remove ivy from my backyard.
A bit. Yeah.
Yeah, the two.
Yes. Yeah. Perfect. Look at that. Amazing. Now the basket star's unfurling. Now that it's uncurling. Well, how about Biobox 2A with the scoop? So we've changed plan because it's unfurled, it won't fit in the quiver.
You know, put it with the net.
Yes, please. There is a basket star called Gorgonocephalus chilensis. And we'll see if it turns out to be that. Yeah, that would not have fitted in a quiver. So, oh, that's great. Thanks, Ben. I think we'll head away from this. We do need to try and find some rocks we can pick up and also some background sediment to core. So let's just see if there's some rocks as we come near the mound that could be picked up. And then we'll move further away and try to push core away from the seep. Was there anything marked about background sediment on Lisa's watch? Is there a waypoint or anything?
Marked for what?
For background sediment?
No, she said to keep on the lookout, but we didn't find it before getting to this location.
Okay.
All the cores are either in the mat or right next to the mat.
Yep. Yep. So we'll just back off. But first we'll look for some rocks just near this mound.
Got you in the station key and all the depth.
So it's on. Like this.
Oh. Yeah. Normal. We just did some studio lighting on basket stars. Getting things back. So in front of you is this. You want to try to find some rocks between here and back off. Less specifically from this guy. Do not open this. There's stuff in here as well. Okay. So,
Thanks, Scott.
People bought by the box for us.
Yeah, thanks.
Okay.
We're just having a pilot changeover. Whoops. No harm done.
Scott.
All right, we've had some fun at the mound, and now we're going to head away a little bit and look for a few rocks. Looking for rocks, and then we're going to do a few push cores. No, we haven't gone looking yet.
Where would you like to go for rocks here?
I think if we just move away from this mound, and hopefully there'll be some free rocks lying on the ground. We didn't really see any rocks we could break off this. Sorry, Doug?
Did you get some good stuff in this spot?
Oh, yeah.
Yeah.
It was great.
I saw that basket stuff.
Yeah. And we tried to make a 3D model. Yeah.
I'm just kind of looking at the interface between the sediment and the...
Yeah, I'm hoping we'll see rocks somewhere here. Could we just have a zoom on this thing?
Yeah.
Some kind of coral, I think. Unfortunately, there's some line that's a bit of a hazard for the ROV. So.
Yeah, can you take my camera and zoom in on this coral and move to see like down side, lower side? Yeah. Grab it out and change it. Go that way.
That's a beautiful orange anemone there, and then next to it an octocoral. It's actually growing over the tubes of a tube worm. Could you grab that on the fly? Sort of avoid that line.
Where are we put?
In a quiver. Please. Yeah, bend open number 1.
I could be wrong, but I thought it was number 4 there.
I thought it was number 4 as
Oh,
well.
was it 4?
Yeah.
Okay. Oh, you're right. Far one. It's 4.
I'll float you in, you clip it, and then I'll float up just to it.
On the whole thing.
Yeah. Perfect.
Nice center over it. Elevation go up to indicate the starboard rail out. What? No starboard rail. What starboard rail? Yeah, not distance. Roger. Good. Can I bring your camera over? Wow. Good.
So these corals will have their DNA sequenced and their anatomy studied to see if they're known species or potentially new. If they're known, it's all valuable to use for range, species ranges. This could have a wide range. Some ocean species can go for thousands of kilometers, others can be endemic to small regions. Great. Thanks, Doug.
Yes.
All right, we'll continue our hunt for some rocks.
No. I think the answer is no, because Mike went to check. I failed on the gas tank all the time. Mike came back and said, we're hopping on the gas tank. Yeah.
So maybe we should make sure it gets put on tomorrow. That's why we put the bubbles that are in the dark line. There's a bit of trash on the bottom here. It's because of the mound, it stops things, and there tends to be an accumulation. We've got darker areas are microbial. The whitish hash is clams. The long tubes could be more of the vestimentiferans, or there's another kind of worm we've seen all over the seafloor that we actually haven't collected yet, a Chytopterid worm. There's a octocoral Anthomastus or heteropolypus there. And purple structure. But we're on the lookout for little rocks that can be easily picked up and put in the bio box. Can we have a close look at these clams as we come up, that white cluster there? Oh, it might be tube worms. Yeah, it's tubes, don't worry about it. Yeah, we can keep going. Looks like a rock. That. Yeah. Can we have the lasers on so we get an idea of how big it is? What do you think? Bit big? Oh, it might fit.
25 centimeters maybe.
Yeah. Okay, we'll give it a go, see if we can get it in. So we're going to try and collect this rock, and it'll be used for geochemistry, but also Lisa Levin, who's along on this journey, will be measuring the rock, how big it is, and then how many animals are associated with it, and what kinds they are. So we've got, it'll be in one of the bio boxes, but we've only got the back 3. So it'll be one of these. But maybe a video, a little close-up. Thanks, Doug. Sorry.
Yeah, no problem.
Yeah.
No problem. So what it does is
Yeah, it's clearing. So we've got a little gorgonian there with a brittle star, snake star wrapped around it. Squat lobster, some hydroids, another little coral in the front. There's a snail there on the left, probably a whelk. And lots of small animals we can't really see. Okay. Let's hope it fits.
Yeah, it shouldn't be stuck to the ground because it's pretty muddy here, right? It's probably 2 minutes tops of that.
Oh, maybe it's a bit big since it's going down into the sediment. Yeah, I think it's too big. Okay, too bad. Okay, we'll keep looking. Yeah, yeah, they were really causing a problem with the mapping, the 3D model.
Is it the
Attracted to the lights. Can we look at that? Might be a rock. With the coral on it like that, trouble is we don't know how big it is. Could be buried. Yeah. That's. Yeah, I think Lisa would want more exposed than that. So let's keep going.
Okay.
Thanks.
Shells?
I don't think that's rock. Oh, this could be some rocks. Or is it just tubes? Yeah. Yeah, this... It's tubes. Nice cat shark there.
Is this too far away from the mountain?
No, we're just looking for rocks. Yeah, it's still pretty active with seepage with these animals living there. It could be a flange. If we could break a piece of it, that would be okay. Yeah, if you could get the manipulator to snap off a piece of that, that would be good. Easier said than done, but it's not too, it doesn't look that thick. There's a bottle. Yeah, but I think if you could break a piece of that, it would be good.
not pushing down very hard. Just 21%. Yeah, you don't need it be more rounded. I'm moving the camera. Yeah, I'm not moving the camera anymore. You're seeing the big waves. Yeah, you're moving it pretty hard. I'm trying to break it. Over a little bit. You tried this with the squash, That's a piece of it, but I don't know.
Yeah. No, no.
More, but do you want that piece or no?
No, thanks, Doug. I think we try for another piece that's a little bigger. There's a thinner piece that might break off easier. I wonder if that might be more thinner.
You can try it, get a crack through it. Where
Yeah.
the bottle is, you mean?
Yeah.
Yeah, maybe.
The stupid bottle in the way.
You just clip the bottle.
Too close to being in there. I'll just try to get it.
So this rock is calcium carbonate. It's generated by microbial activity, which is pretty amazing, really, to find so much rock down here. But it's all because of this methane that's seeping up through the seafloor, and then the action of bacteria and archaea working together to break it down. And that results in carbonate also being formed. And then that, oh, very nice. Perfect.
Float upside over the drawer.
Maybe a nice grab of it. I don't want to stop you before with it dropping, but we'll try and get a picture. Okay, good enough.
Alright, you ready for me to push out? Okay.
It can be in box 1 or box 3. Box 2 has something on the port side.
Now to retract the camera, right?
Yeah.
So permanent cover. Alright, so put the camera in here. Okay. Box 1.
Yep, box 1, any side. Great. So that's B1B.
That was BB1B.
BB1B. Rock 1. See if another one there is possible would be good.
Another rock?
Yes, please.
Okay. We'll go back down. No poor shower managed access. Conduct. We want another shower. Okay. I think we're either right next to where you see it goes, or just a little bit to the left there, right? That's.
So there's not so many obvious seep animals here. We've got quite a few little corals, octocorals, the pink branching things you can see, hydroids, some squat lobsters, some shrimp. I'm just looking for another flange that maybe can be broken off. Oh, perfect. Yes, please. Yeah. Actually, a nice grab of that cut face. It would be interesting. Look at that.
Wow.
Well, the geologists will be excited to subsample this.
Scott's rock box. Those look like clams.
Could be old clam pieces in there with the carbonate accreted around them.
There's bubbles coming out.
Yeah, that's great. Okay.
Yep.
Rock 2.
Even me.
Same box?
Yes, other side, please.
On 1A?
Yes, please.
Okay.
You ready for me to push up? Okay, come on. Pull the camera back in. Right there. Pushes out. Box 1. Here we go.
Oh, great. Thanks, Joe. Lisa will be happy. But we'll hopefully get some more. But I think we might want to head off-site a little bit, away from the mound, and see if we can get some background sediment for coring.
Okay, I'm gonna take the opportunity to swap over the stream to part 2. If you're on Facebook, part 2 will be up shortly. And if you're on YouTube, you should be redirected to part 2 automatically.